Hi students. Am I audible? Yes, am I audible, visible? Hi students, am I audible? Am I audible? Yes. Good evening, good evening, good evening. Am I audible or not? Am I audible or not? Hi students, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. I hope you guys are doing very well. Hi, do let me know in the chat section, am I perfectly audible and visible to you all? Am I perfectly audible and visible to you all? Yes? Yes, great guys, great. So welcome to the session students, and today we are going to discuss this chapter, that is Animal Kingdom. Again, from Animal Kingdom minimum 4 questions are expected. Okay, 4 questions are expected from this particular chapter. And when we talk about this chapter, guys, basis of classification should be clear to you. Right, you should know about the basis of classification first. Then please try to memorize the name of all the phylums in the same flow, right? In the same flow, right? In a particular flow, right? The way it is given in NCERT, you people should memorize the name of the phylums in that flow, and trust me, it will help you to solve at least 2 questions from this particular chapter. At least 2 questions from this particular chapter. Okay? Okay. So are you ready to start and to finish this particular chapter? Do let me know in the chat section, guys. Are you ready to start and to finish this particular chapter? Yes? If it is a yes, I want to see some fire emojis in the chat section, guys. Quick. Are you all ready to finish this chapter? Yes, I know some students, they hate this chapter. They hate this chapter because there are so many examples that they need to remember, right? But students, obviously, we do not have any choice. We do not have any choice, and yes, we have to master it, right? We have to master it. We don't have any choice, but it is simple as well. See, if you have seen my last video, right, in which I discussed about the biology strategy, so in that particular video I mentioned it. Right, in that particular video, I mentioned it specifically, right? I mentioned it very specifically that when, like for the memory-based topics, right, you have to give your 30 minutes every day to remember that. If you are thinking, nahi yaar, I'll do it in the month of February, I'll do it in the month of March. No, it's not going to help. Literally, it's not going to help. Give your 30 minutes every day. For the plant kingdom, we have the tricks, right? And in animal kingdom, a lot of examples are there, and then you have to remember a lot of tricks. So it's better to read these phylums and to remember the name. And trust me, it's all about the practice, right? Trust me, it's all about the practice, right, bache? Right, bache? Okay? Okay, chalo. So let's start the chapter. But before that, let me tell you guys, and I hope you all are aware of it as well. See this batch, right? So our Phoenix Master batch is live, and in biology we are about to start class 12th. Okay, so I can say from 1st of December we'll be starting class 12th syllabus along with class 11th revision. So without any delay, be the part of this batch, right? And your complete team Avengers is going to teach you there. And trust me, the batch is amazing, right? And you will get a proper direction, okay, if you will enroll in this particular batch. So in the description box there is a link, go for it. And now I'm going to start the chapter. Okay, so the chapter name is Animal Kingdom, right? The chapter name is Animal Kingdom, right, bache? Animal kingdom. Okay, chaliye, let's discuss about this particular chapter. Okay, let's discuss about this particular chapter. So are you ready for that? Yes. Are you ready for that? Are you ready for that? Animal kingdom. So students, I will start from the basics part. You know that R.H. Whitaker, he is the one who gave us the 5 kingdom classification system, right? He is the one who gave us the 5 kingdom classification system. Yes or no? Tell me, yes or no? He was the one who gave us, right bache? He was the one who gave us what? Who gave us the 5 kingdom classification system, isn't it? Isn't it? Yes or no? So when we talk about these 5 kingdoms, you know that we start with the kingdom Monera, then we have kingdom Protista, right students? Then we have fungi, Plantae and Animalia. These are the 5 kingdoms that we have, right? And obviously there are some general characters, right? On the basis of that, we place the organisms in different kingdoms. Like if we belong to animal kingdom, obviously there are certain characters that we need to fulfill. That's why, right? That's why we are in this particular. That's why we are in this particular kingdom. That is kingdom Animalia. Okay, that is kingdom Animalia. So when you talk about this kingdom Animalia, one thing is for sure, that all the organisms are going to be eukaryotic here, right? That all the organisms are going to be what? All the organisms are going to be eukaryotic here, right bache? All the organisms, they are going to be eukaryotic here. We are talking about what? We are talking about the kingdom Animalia, right? Plus here you are going to see multicellular organisms. What kind of organisms are we going to see here? We are going to see multicellular organisms. And here, right, obviously there are different phylums there. So we will see that how do we move from the level of simplicity to complexity. Okay, okay. Right, from simplicity to complexity we'll see, right? We'll start with the Phylum Porifera, then we will talk about the Phylum Chordata, and we will see that we will move from the level of, from simplicity to the complexity. So when you talk about the organisms here, definitely organisms are going to be multicellular. Without any doubt, they are going to be multicellular, right? Without any doubt, they are going to be multicellular, but here we will see that there will be different level of organization, right? It means that some organisms, they will just have cell in their body, right? They will just have cellular differentiation in their body. Some will form tissue, some will form organ, and organ system will be there. That's how it works. Okay, that's how it works. Is that clear? That's how it works. Is that clear? Is that clear? Is that clear? So that's what we need to study in this particular kingdom, right? Which kingdom it is? It is the animal kingdom. That's what we have to study in this particular kingdom. Okay, bache. So when we talk about this classification part, yes, we discuss 7 broad categories, right? I hope you all remember this, that we talk about the 7 broad categories. That's what we discuss, right? 7 broad categories. Yes or no? Right? So you know the trick for that, that is keep pot clean or family gets sick. Keep pot clean or family gets sick. Keep pot clean or family gets sick. Right? So kingdom is there, phylum is there, class is there, order is there, family is there, genus is there, and then what do we have? We have the word species. That's what we have. These are the 7 broad categories, right? So these are the 7 broad, or we can say that 7 obligate categories, right? Obligate categories. But students, we also have like this is the kingdom, right? In these kingdoms, we have the sub-kingdoms also, right? In these kingdoms, we have the sub-kingdoms also. They say when you talk about the kingdom Animalia, right? When you talk about the kingdom Animalia, it is divided into 3 sub-kingdoms, right? Do you know the name of that 3 sub-kingdoms? Yes. Do you know the name of that 3 sub-kingdoms? Yes. Tell me. Do you know that? Please tell me. Right? Do you know the name of that sub-kingdoms? Yes, bache? Yes. Right? So we have the kingdom Animalia. What do we have? We have the kingdom Animalia, and it is having the subkingdom. So when we are talking about the kingdom Animalia, we will mainly talk about two subkingdoms, right? Mainly here under this kingdom Animalia, we will be talking about two subkingdoms. That is your subkingdom, right? I'm not going to discuss it in detail, but please pay attention. That is subkingdom Parazoa. That is subkingdom Parazoa, and there is one more subkingdom, right? There is one more subkingdom, that is your Eumetazoa, right, bache? Eumetazoa. Do you know about this? Do you know about this? Actually, I'm going to discuss it is not something very important that you need to note down in your notes, but you please try to understand it. Okay, bache? You please try to understand it, right, bache? Right, bache? Yes. So what are we talking? We are talking about two subkingdoms here. The subkingdom Parazoa and the subkingdom Eumetazoa is there. See, overall, if we follow the NCERT part, right? Overall, if we follow the NCERT part, even in NCERT, when you talk about the phylums, and please remember this thing. As I said in the starting, you need to, right? As I said in the starting, you need to remember the name of the phylums in that in a particular order. Like let's start from Phylum Porifera. Let's start from Phylum Porifera. Then we have Phylum Cnidaria, also known as Coelenterata. Ctenophora is there. Platyhelminthes, also known as flatworms. So if you find it difficult to remember, you can just focus on the first 5 names first, right? But you can just focus on the first 5 names and then repeat the next one. So like you can even write it on a paper. Okay, so Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes. Porifera, right? Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes. Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes. These are the first 5 names. So Platyhelminthes means your flatworms, Aschelminthes your roundworms. Right, these are the nematodes basically. Okay, after that we have Annelida, right? After that we have Annelida. Then Arthropoda is there, largest phylum of animal kingdom. Then Mollusca is there, second largest phylum. Then it is Echinodermata. Then it is what? It is Echinodermata. And then we have Hemichordata and Chordata. Hemichordata and we have Chordata. So do you remember these names? Yes, yes, please. Do you remember these names? Please tell me. Do you remember these names? Please tell me. Yes. Do you remember these names? Okay, so what are we talking? We are talking about the phylums here. So these are the 11 phylums that we have to discuss here. So we are starting with the Porifera, Cnidaria, Ctenophora, Platyhelminthes, right? Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Right? These are the important phyla here. Right? These are the important phyla here. Yes or no? Yes or no? Tell me, these are the important phyla here, right? Bache, 11 phyla. Okay, now what am I saying? See, we are talking about the animal kingdom, and we know that multicellular organisms, eukaryotic organisms are going to be there, right? Bache, they will have different, like, here when we talk about the phyla, we will move from the simplicity. When we talk about all these 11 phyla, we'll move from the level of simplicity to the complexity. We will see organisms are having primitive. We'll start from the primitive characters, basic characters, of course, right? That's how we are going to move, and that's what we need to discuss. Then even today, I told you that there are subkingdoms also. Are we going to discuss that in detail? No. I will just give you one hint, right? I'm just trying to relate it with the biological classification part as well. So we have the subkingdom Parazoa here, and here we talk about, under this subkingdom, we talk about Phylum Porifera, right? So your Phylum Porifera, your Phylum Porifera. We start the discussion from Phylum Porifera, right? That Phylum Porifera, porifera means pore bearing organism. Pore bearing organisms. Here we talk about the pore bearing organisms. They are having that pores, right? Sponges. We are discussing about the sponges. We'll discuss about that in detail. So there is subkingdom Parazoa. So now when you talk about this subkingdom Parazoa, students, now when we talk about this subkingdom Parazoa, here you will see organisms. They are having a multicellular body. Like multicellular body is there. Tissue is still not there. Tissue is still not there. You know now how it starts? Cell, group of cells, tissue, then organ system, then organism. But here you are not going to see, yes, here you are not going to see what? You are not going to see the tissue level differentiation. Here in the organisms you are not going to see the tissues, but cells will be there. Plus it's a group of cells, like group of cells having, you know, some canals, having flagellated cells. Right? Matlab Parazoa, under this we include what kind of organisms? We include the organisms which are definitely multicellular, which are having some canals, right? Having some flagellated cells, right? Cellular differentiation is there, but tissue, right? Tissue level of differentiation is still not there. Are you getting my point? Tissue level of differentiation is still not there. Right, bache? Right, bache? Okay, so the cells here, they will function more or less independently. They will cooperate with each other a little, right, bache? So tissue differentiation is absent, but cellular cell differentiation is definitely there, right, bache? Cell differentiation is definitely there. Is that clear? Is that clear? So this subkingdom will include a single major phylum, that is your Phylum Porifera. And here we see that organisms, even they are immobile. They are not movable. So if I'm saying immobile, sessile is the word. Sessile. They will be attached to the substratum. They are not moving. They are not moving. That's what we discuss here, right? That's what we discuss here. Actually, we, you know about the protozoa, right? Kingdom Protista. Kingdom Protista is having protozoans, right, bache? So somewhere you can say that from protozoans you are moving here, right, bache? Some connecting structures are there in between, some multicellular flagellated structures are there in between, and then we get the porifera. So I'm not discussing it in right, I'm not discussing it in too detail, but you should know this. So Phylum Porifera is coming under this subkingdom. Rest all will be there in Eumetazoa. And when we are talking about this Phylum Porifera, yeah, in general you can say that study of poriferans. Study of poriferans is what? It's parazoa, so it's parazoology. It's parazoology. So here you will see the simplest kind of organisms that are not even, right? That are not even looking like animals, because they are sessile. They are attached to the substratum. And even, you know, when taxonomists, they were searching the things, you know, so even these organisms, they were not even kept under kingdom animal, right? Later on, when their development was studied, then it was like, okay, they are animals, they are not plants, they are animals, and then they were kept under kingdom Animalia. Okay, then they were kept under kingdom Animalia. Okay, okay, got it, okay. Done, bache? Okay, so that's what we need to discuss. So see, different types of, matlab you look around, even at your home, you can see lizards are there, flies are there, mosquitoes are there, cockroach, spider, right, bache, dogs, right, different types of animals are there. Humans are also there. We all are so different. Just compare yourself with a lizard. We are so different from each other, but still we are kept in a same kingdom. So no doubt we have the differences, but there are certain general characters also, right, that we all share, right, that you can say that are our similarities, right. There are some general characters also, and we have the differences also. So overall, even in the chapter living word, that's what we discuss, right. I don't know whether you know it or not. See, when you move from kingdom to the species level, when you go down, but specific characters they increase, general character they decrease. And when you move from the species to the kingdom level, general character, like specific character, specificity will decrease. Now what's the meaning of that? All the organisms, just I'm saying, you compare yourself with a lizard, you compare yourself with a sponge, right? We are so different from each other, still we belong to the same kingdom. The reason is certain characters that we have. They are the general characters. Now, we are in phylum Chordata, right? We humans, we are in phylum Chordata. We are under subphylum Vertebrata, right? We are under class Mammalia, right? So we share more similarities with the mammals. We are not sharing that much similarities with the arthropods or with some other organisms. No, with the organisms that belong to the same class. Then obviously you will say same order, family, like this, right? Like this we have the things, right? So now what am I trying to say? Here, right? Now, see, listen to me. Right? So what am I trying to say here? I'm saying that we all are kept in this kingdom on the basis of certain characters, that are the general characters. So when we study the phylums individually, when we study the organisms under different phylums, so obviously on the basis of certain characters, you say we belong to phylum Chordata, we have certain characters. Insects are there in phylum Arthropoda, they have certain characters, right, bache? On the basis of that specific characters, we are kept in different phylums. It's like that, right? It is like that. Are you getting my point? It is like that. Are you getting my point? So here in this particular chapter, we will start our discussion from basis of classification, then we will study these phylums individually, and now onwards I will discuss the things as per the NCERT only, and only few characters or few things which are important for your NEET, right, and which are there, which are as per your PYQs, that things I'm going to discuss, rest nothing, okay? Rest it will be as per NCERT. So this basic introduction clear, so the name of the phylums clear, yes, so the name of the phylums clear, tell me. Yes, name of the phylums clear, sure. So repeat it with me. Start with the Phylum Porifera. Write down the name of all the phylums. Right? Write down the name of all the phylums. All the phylums here in the chat section, guys. Quick. Quick. Write down the name of all the phylums in the chat section. Yes, please. Write down name of the phylums. Very good, Sai bache. Yes. What about others? Done? Chaliye, let's start it. Done? Chaliye. So let's start with the basis of classification. Right? Let's start with the basis of classification. Clear, bache? Let's start with what? Let's start with the basis of classification. Right? Let's go for that. See, when you talk about these bases of classification, what are we going to discuss here? We will start with the level of body organization. What are we going to discuss here, bache? We will talk about the level of body organization, right? We are going to discuss the symmetry here. Yes, we are going to talk about the germ layers here, right? We will discuss about the coelom here, right? And we will talk about the segmentation here. We will discuss about the body plans here. We will talk about the notochord here. So there are certain characters. On the basis of that, we classify the organisms into different phylums, right? On the basis of that, we classify the organisms into different phylums. So let's start with the level of body organization, right, bache? Let's start with what? Let's start with the level of body organization. So when you talk about the level of body organization, right, if I start from the basics, right, if I start from the basic things, just say for an example, you know that firstly we have a cell, right? Firstly, what do we have? We have a cell, and that cell is having what? That cell is having what? Right, in the chapter cell we have discussed that. In the chapter cell we have discussed this thing, right? In detail we have discussed this thing. Okay, in the chapter cell, in detail, we have discussed this thing. And what is that? That cell is the basic unit of life. Below the level of cell, there is no independent existence, right? The organelles cannot exist by its own. Whatever it takes to be a living, a cell is having everything. And there we talk about the protoplasm. There we talk about the protoplasm, which is also known as physical basis of life. Right, it is the physical basis of life by Huxley. Huxley said it as physical basis of life. You are talking about the protoplasm. What are we discussing here? The cytoplasm and the genetic material, isn't it? The cytoplasm and the genetic material, right? So this protoplasmic level of organization is there at the first place, and that we see in the unicellular organisms. That we see in the unicellular organisms like your protista, right? Like your protista, like your protozoa, right, bache? So in these unicellular organisms, all the life activities will be there for sure. Now in the kingdom Animalia, in your subkingdom Parazoa, in your Phylum Porifera, right, you will see only multicellular organisms will be there. So when you talk about this level of body organization here in animal kingdom, you will talk about the cellular level of organization. Yes, we will talk about the cellular level of organization, right? We will talk about the tissue level of organization, right, bache? Organ level of organization, and we will have organ system level of organization. We will have organ system level of organization, right? Cellular level is there, tissue level is there, organ level is there, organ system level is there. Are you getting my point? What do we have here? We have the cellular level here, we have the tissue level here, we have the organ level here, we have the organ system level here. Are you getting my point? We have the organ system level here. So when it is the cellular level, right, just say multicellular body only a multicellular body. Only a multicellular body, right? So you are talking about the poriferans here, porifera. So in the phylum Porifera, all the organisms, they just have cell, as I said earlier. They do not have the tissue level of differentiation. Now, there are the organisms. There are organisms, right? So when I'm saying porifera, we are talking about the sponges. We are talking about the sponges. Now, tissue level of organization. These cells are making tissues. Means now we have the animals. Now we have the animals in which the differentiation is there at the tissue level. Cells together will form the tissue, but organs are still not there. Organs are still not there. Here you talk about phylum Cnidaria, and you are going to discuss about the phylum Ctenophora, also considered as a minor phylum, right? So these two phylums are there. That's why I told you to remember these names in a sequence only, in a proper order only. So Porifera, Cnidaria and Ctenophora, tissue level of organization. So if you don't know the example, Cnidaria, Hydra is there. Hydra is there. Or you can say your jellyfish, Aurelia. Okay, Ctenophora mein, Ctenoplana. There are different examples. We will discuss it one by one. Then organ level of organization, right? So now in these organisms, you will see organs are there. Organs are there. You have Platyhelminthes, helminths, flat worms. Their body is dorso-ventrally flattened. Then bache from the phylum Aschelminthes, from the phylum Aschelminthes to Chordata, they all are having organ system level of organization. But one more thing I would like to specify here, it's biology, science of exceptions, right? Bache, science of exceptions. Even in this particular chapter, that is your animal kingdom, let's say we are talking about the phylum Porifera, they will give us the general things, right? Just say fertilization is this, the sexes are this and that. But individually, when you study the different organisms there, like whatever characters they give us, right, it is on the basis of what? On an average, that's what we are going to find, right? Like majority of organisms are going to have that, right? But yes, exceptions will be there. Exceptions will be there. Okay, okay, so these things are literally, they are vast. Okay. Done, bache? Yes. One second, students. Done? So this is the first thing, right? Next thing is the symmetry. Next thing is what? Symmetry. Now you tell me, what do you understand with the word symmetry? Like in general, I'm not saying explain it in the terms of biology. In general, what do you understand with the word symmetry? What do you understand with the word symmetry? Yes, what do you understand with the word symmetry in general? Please tell me. In general, I'm not saying ki explain it in terms of biology. We will discuss that. Like when we are saying something is symmetrical, cutting in body plane, acha shape, ya division, okay, anything else, divide into two parts. Matlab, is it about the two parts only? Is it about the two parts only? When we are saying, like say there is an organism which is having similar in shape, right? It is having some similarity in shape. It is having some similarity like in position, like on various aspects we can consider that, right? On various aspects we can consider that, right? It's not like you can say that not in one context we are talking about it. In general, when we are saying symmetry, let's say something is balanced, something is balanced. It's like that. It's like that, right, bache? Right? So in terms of maths, in different terms, right, we can define the symmetry thing in a different ways. Now in biologically, what do we say when we say an organism is symmetrical, right? What do we consider when we say an organism is symmetrical? So when we are saying symmetry is there, something which is having similar shape, right, something like that. Now here, when we are talking about the symmetry, let's take an organism. Like let's see, it is some structure, can be something else. Ginger, let's take the example of a ginger. Like it is having different shapes. Like you pick one ginger and compare it with another, right? You cannot say both the structures are ditto same, right, bache? Right? You know that the way it grows. Try to visualize the thing, try to relate the things, okay? Try to relate the things. Now, I'll give you another example of a starfish. So when we see a starfish, you know that we can divide it into equal halves. Right, bache? So what am I saying? I'm saying that if we have a structure that we can divide, right, that we can divide into equal halves by one way or another way, we'll say it is having some symmetry. It is having some symmetry. But if it is not having that, then it will be asymmetrical. Just say in the animal kingdom also, right? In the animal kingdom also, the next character that we consider is symmetry. So we have asymmetrical organisms also. What kind of organisms do we have? We have asymmetrical organisms also. Asymmetrical, without any symmetry. They do not have any symmetry. We cannot divide them into equal halves. Right, we cannot divide them into equal halves. They are asymmetrical. Can you quote any example here? Right, when we are talking about the symmetry, right, I'm saying some organisms, they are asymmetrical. Any plane that is passing through the center cannot divide this body into equal halves, right? When any plane that is passing through the center, cannot divide the body. Yes, when any plane that is passing through that center cannot divide the body into equal halves, right? Then it is the organism, or it is asymmetrical organism. Are you getting my point? It is the asymmetrical organisms. Asymmetry it is. Are you getting my point? So, jaise we have Phylum Porifera, mostly poriferans, right? Mostly poriferans. Again, I'm saying mostly, I'm not saying all the poriferans. Mostly poriferans are asymmetrical. They are without any symmetry. We cannot divide them into equal halves. When the plane passes through the center, it cannot divide them into equal halves. But again, we have the exceptions also. Just say you have the example of Scypha, right? You have the example of Leucosolania. These are the poriferans, but they have radial symmetry. So what do they have? They have radial symmetry. Are you getting my point? Right. Now, this is the First thing, asymmetry. Then comes the radial symmetry. Now, what is the meaning of radial symmetry? So, try to remember it with the help of examples, right? Try to remember it with the help of examples. Just say, let's say, starfish. Starfish. Yes, we can divide the starfish into two equal halves. We can divide the starfish into two equal halves. So, yes, starfish is symmetrical. Starfish is what? Starfish is symmetrical. But the point here is, right, that how can, like, which plane is going to divide it into two equal halves? So students, right, when we are saying radial symmetry, so you will say when any plane passing through the center, when any plane, please write down, when any plane passing through the center, of body axis, when any plane passing through the center of body axis, see, imagine the starfish, okay, it can divide the animal into two, it can divide the animal into two equal halves, or two identical halves rather. So we will say organism is having radial symmetry, right bache? Right bache? So any plane, the word is any plane here, but here we were saying when any plane passing through the center of body axis cannot divide. Here, any plane passing through the center of body axis cannot divide the body into identical halves, but here it can. So you have the radial symmetry. What do you have here? Radial. So after Porifera, you have Cnidaria. They are having this radial symmetry. Ctenophora, they are having this radial symmetry. But one more thing is there, bache, adult echinoderms. Just this starfish. Starfish belongs to your phylum Echinodermata, right? Starfish belongs to your phylum Echinodermata. So adult echinoderms, okay, adult echinoderms, they have radial symmetry, right? Adults are having radial symmetry, but students, but larva, right? Larva, they have bilateral symmetry. They have bilateral symmetry, and it is basically related to the evolution part, okay, right? So remember this, adult echinoderms, but larva is bilateral symmetry. But larva is having bilateral symmetry. Now the question is, what if in the paper they ask us, right? What if in the paper they ask us about the symmetry in the echinoderms? So remember, because adults, right? Because adults, they possess the radial symmetry. So overall we say this phylum is having radial symmetry. But if they are specifically talking about the larva, then we have to mention the bilateral symmetry. This part clear? Then we have to mention the bilateral then we have to mention the radial symmetry. This part clear? Yes, this part clear? Right? So next is bache bilateral symmetry. Next is what? Next is your bilateral symmetry. Next is what? Next is your bilateral symmetry. So see, bi means two. Bi means two. Lateral means sides. Bi means two. Lateral means sides. Right? Two sides. So when, see, when we can divide the body into two identical left and right half. Right, here we are being specific. Here we are saying when any plane. But here, when the body can be divided into, identical right and left half only by one plane, right? Only by one plane. Then we will say it is bilateral symmetry. Right? So when the body can be divided into identical right and left halves in only one plane. Okay? In only one plane. Yes, when the body can be divided into identical right and left halves right, when the body can be divided into identical right and left halves only by one plane is your bilateral symmetry, right? Is your bilateral symmetry. Is that clear? Is that clear? So here you will talk about the platyhelminths. right bache? You will talk about the platyhelminths. See, after your this Ctenophora, we have the platyhelminths. So platyhelminths to chordata we will have, right? So again, here you have to remember that echinoderms, adult echinoderms, radial symmetry, but larva is having bilateral. Plus in mollusca, one student is asking me this. In the case of mollusca, bache, some mollusks, they are secondarily asymmetrical. They are secondarily asymmetrical due to torsion that we will discuss. Okay, when I have patience, that we will discuss. Right, bache? Right. That we will discuss. Okay, so you guys can see the examples here. When any plane passing through the center can divide the body into equal halves, radial symmetry. When body can be divided into identical left and right halves, right, in only one plane, right, then it is the bilateral symmetry. So please check this picture. Please check this picture. Yes. Okay, chaliye. So next, let's move to the next part. See, this diagram is from the NCERT. So this is the radial symmetry. This is the bilateral symmetry. Okay. So this is about the symmetry part. Now, the next thing that we need to discuss is, germ layers. And after that, see, in the NCERT, you know that open circulatory system, closed circulatory system, complete digestive system, incomplete digestive system, everything is given. We'll discuss it. But after completing this major part, okay, then we will talk about the body plans. Okay, right? So now the next thing is, bache, germ layers. What are these germ layers? What are these germ layers? Yes. What are these germ layers? See, when we talk about the germ layers, students mostly they answer like this: diploblastic, triploblastic. What exactly is that? What exactly is that? Let me tell you. See, you know that we all develop from a single-celled zygote. We all develop from a single-cell zygote. Yes or no? We all develop from a single-cell zygote. Yes, you know that. You know that this zygote is a totipotent cell. What is this zygote? This zygote is a totipotent cell. Okay, what is it? It is a totipotent cell, right? Totipotent cell means this cell is having the ability to develop into a complete organism. Now you know that, like if I talk about the mammals, okay? If I talk about the mammals are having the slowest, right? Mammals are having the slowest cleavage of the slowest cleavage of the animal kingdom. Means this zygote, you can say after 30 hours this zygote will divide. It will form two cells, two blastomeres. Okay, it is going to form two blastomeres. So we know that we all are developed from this single cell zygote, and we are a multicellular body. We are made up of many cells. So this single cell organism, if it has to make a multicellular body, it has to divide. It has to divide and divide. right? It has to divide, divide and divide. So this single cell will become, right, the single cell, firstly it will divide. Just say I gave you the example of mammalian cleavage. After 30 hours it will divide. You will get 2 cells, then 4 cells, then 8, then 16, like this. Like this. So basically when we talk about this development now, we start from the zygote and then slowly we will see how the embryo, then the fetus, then the complete organism forms. So basically this zygote, it will divide from single cell, it will become multicellular structure. So during this development now, student, you will see different stages. Just say you will talk about morula, you will talk about morula, you will talk about blastula, you will talk about gastrula also. Gastrula also, like these are the different stages of development. Like when I'm saying morula na, you will see that there is a ball-like structure having some cells, ball-like structure. When I'm saying blastula, blastocyst, it will have the cavity, it is having the cell. This ball-like structure, more cells will be added, further development will be there. So here, right, when we talk about this development, you will see how the single-celled organism, how the single cell will divide, will become multicellular, plus we will see how that cell layers, they will move, they'll show some movements, right? We will see that these cell layers, they will show some movements. We call them as morphogenetic movement, right? We'll call them as morphogenetic movement. Some movements will be there, and that multicellular structure, right, that multicellular structure, slowly that cell layer, right, because of some movements, I'm not talking about that movements, I'm trying to explain this thing. You people try to visualize the things. So that multicellular structure, some movements will occur and it will result in the formation of different layers. So when I'm saying gastrula, right, or gastrula formation, that is gastrulation, we will see some layers will form. We will see some layers will form. We call them as germ layers. We call them as germ layers, and they are also referred to as embryonic layers, right? So germ layers, germ to germinate. That's how you can remember. Germ to germinate. Actually, these layers, they can form the different body organs. So basically, just imagine a single cell, then it is dividing into 2, 4, 16, it is becoming multicellular. So during its development, we will see sometimes some cell layers, some cell layers, they will move in such a way, they are going to form some germ layers, right? These germ layers are responsible for the formation of different body organs, different body systems, right? Different body system. So when you talk about these germ layers, on the basis of that, when we classify the organisms, so we will see we have two things: diploblastic organisms, and we will see that we will have triploblastic organisms. So when we are saying diplo, di means 2, tri means 3, right? You are talking about the germ layers. Actually, students, 3 germ layers are there: ectoderm, mesoderm, and then endoderm. Outer one, ecto, mane outer, endo means inside, M for mesoderm, M for middle. These are the 3 germ layers. So the organisms in which we will see 2 germ layers, that is ectoderm and endoderm, they are known as diploblastic organisms, right? Because 2 germ layers are there. Outer ecto, inner endo. So they have 2 germ layers, diploblastic organisms. So the organisms, right, in their development, if they have 2 germ layers, they are diploblastic. And if they have 3 germ layers, that is your ectoderm, middle one is mesoderm, and then you have endoderm, then it will be, that organisms will be triploblastic organisms, right? That organisms will be triploblastic. So now I'll give you one example. Just say if you talk about the ectoderm, in our case, nervous system, ectoderm will form the nervous system. Your muscular system, your skeletal system, who's going to form that? Mesoderm. Again, exceptions are there. Your digestive system, your gut lining, your endoderm is going to form that. So these layers will form our different systems. These layers, they will form our different systems. So we have diploblastic organisms if 2 germ layers are there. We have triploblastic organisms if 3 germ layers are there. Okay, if 3 germ layers are there. Now, one more thing that you need to understand here. And that is, just see, let's say you are taking a section of an organism. Let's say you are taking a transverse section, right? You are taking a transverse section. So this one, this outer one is ectoderm, right? This inner one is endoderm. Okay. Now, this is the gut cavity. We'll come to this. We'll discuss that too. So, this layer, let's say cellular, it is having the cells, right? Ectoderm and endoderm is there, right? What do you have? You have the ectoderm and you have the endoderm here. Now bache, in the diploblastic organisms, we will see that in between this ectoderm and endoderm, we will see that there will be undifferentiated layer. Matlab it is not made up of cell. We will see something in between. We call it as mesoglia. And it is undifferentiated. It is not made up of any cell. It is undifferentiated. Just say ectoderm is having cells, endoderm is having cell, and in between you have mesoglia, an intervening undifferentiated structure. So that's what you are going to see in the case of diploblastic organisms. So again, outer one ecto, inner one endo, right? Outer one ecto, inner one endo, and in between undifferentiated mesoglia, diploblastic organism. So here you will see your coelentrates, your coelentrata, your ctenophora. They are diploblastic, right? And from phylum Platyhelminthes to chordata, all the organisms, they are triploblastic. Right? Now, what about porifera? Bache, poriferans, like in some books, yes, it is mentioned that they are diploblastic, but overall, there is no clarity related to that. Poriferans, they are considered as the one that are without a germ layer, right? They don't have germ layer. Is that clear? So now, if our organism, they are triploblastic, then what will be the scenario? This will be the scenario, right? So look at it. So this is your outer ectoderm, this is your inner endoderm, and this is your mesoglia. Right? What is it? This is your mesoglia, right, bache? This is ectoderm, this is endoderm, and this is your mesoderm. Is that clear? So this is ectoderm, this is mesoderm, and this is your endoderm. Clear, bache? So that is what we have. So these diagrams should be clear, right? These diagrams should be clear. So about the germ layers, everything clear? Now, bache, we will talk about the next basis of classification, that is the coelom, the body cavity. So tell me, what do you know about the coelom, about this body cavity? Yes, guys, stop discussion and please answer it. Yes, what do you understand? Coelom means space. That's it, Tej. Anything else? Region between body wall and gut wall. Anything else? Whatever you know related to the coelom, just let me know in the chat section, guys. Let's make the session interactive. And if you are new to the session, just quickly hit the like button. Yes. Coelom large. Okay, देखिए बच्चे, let's start from the porifera. Okay, let's start from the porifera, like your sponges. I have seen many students sometimes they ask me this thing, even in the sponges we have the cavity. This is sponges may, when we talk about their internal structure, we say that they are having a cavity in between. We call it as gastrocele, right? The water will flow through it, then water will come out. We discuss that, but still we say that poriferans, they are acoelomates, right? They are acoelomates. So what is the meaning of being acoelomate? Acoelomates means without body cavity, yeah, without coelom. Coelom, you are right, it is the body cavity, but here also we have the coelom, here also we have the cavity, but still why do we say it is without coelom? I have seen student asking this thing again and again. See, under this character, under this thing, what we need to discuss, we will talk about the acoelomates. Remember, we will talk about the pseudo-coelomates, right bache, and we will talk about the true coelomates, that is your eucoelomates. These are the three things that we need to discuss. Yes, you are right, coelom mane body cavity, right? Yes, you are right, coelom means body cavity. Now try to understand it. Let's try to understand it. Even in the poriferans, there is a spongiocele, right? Even in the poriferan, there is a spongiocele here. Then why do we say that they are acoelomates? They are without any cavity. Now let's understand what exactly is this coelom, right? Let's understand what exactly is this coelom. Okay? Chalo, let's go for it. Okay, so I will start with the body plan thing here. Okay, I will start with the body plan thing here. I will start with the body plan thing here, right? How will I start? I will start with the body plan thing. Chalo, what is the meaning of that? Dekho, when you talk about the body plans, one is cell aggregate body plan. One is cell aggregate body plan. So what is the meaning of cell aggregate body plan? That there is a multicellular body, that's it, that's all, right? That there is a multicellular body, that's it, that's all. Okay, so what am I saying here? I'm talking about the body plan. I'm saying cell aggregate body plan, and you know that we see in the case of sponges. Then bache, we have blind sac body plan. What do we have? We have blind sac body plan. Blind sac means something which is having one opening and the other end is closed. Something that is having one opening and the other end is closed. Again, I'm repeating, something having one opening and the other end is closed. Now, from this opening is like a mouth, as well as it is like an anus. Means from this mouth, from this opening, ingestion will also occur and egestion will also occur, right? So when you see such kind of arrangement where one side is open and the other side is closed, blind sac. Now let's say if any organism is having that type of body plan, right? So that organism is having blind sac body plan, and when you talk about their digestive system, it is incomplete kind of digestive system. It is incomplete kind of digestive system. This digestive system is incomplete, right? This digestive system is incomplete, right? Because same opening is serving as a mouth as well as an anus, right? The same opening is serving as a mouth as well as an anus. Blind sac body plan. Then there is a tube within tube body plan. Then there is a tube within tube body plan. I'll come to that coelom thing also, but let me complete this part. Tube within tube. Now here you will see two different openings. One will serve as a mouth, and another will serve as an anus. Okay? So when you will see such type of tube, right, that means it is the complete digestive system. It is the complete digestive system. What is it? It is the complete digestive system. Now, the organisms that are having this, firstly, they have this tube, they have this gut tube, they have this tube where one mouth or one anus is there. Plus, they are also having the outer body wall. So can I call it as tube within tube body plan? Can I call it as, let's compare outer covering as a tube and inside there is also that gut part, right? So tube within tube body plan. So obviously it means the digestive system is complete. So when you are saying blind sac body plan, you are talking about the coelentrates. From coelentrates to platyhelminthes, their organisms are having blind sac body plan, and rest of them are having Yes, bache. Rest of them are having tube within tube body plan. Right. Now, ma'am, we were talking about the coelom, and now suddenly you switch to this part. Why? I'll tell you. Now, just imagine this is the case. This is the thing. Right. Just imagine this is the case. This is the thing. There is the outer body wall. There is an inner gut wall. Right. Outer body wall is there, and there is an inner. This inner gut wall is there. Right. Right, basically gut wall. I'm talking about the alimentary canal here. So if in between these two, just let's say this is your gut wall, this is your outer body wall. If in between these two, if there is a space, right, if there is a space, plus that space is also lined with mesoderm, Again, I'm telling you, just try to imagine this. You have a tube within tube body plan. So in tube within tube body plan, I'm saying consider the outer body wall, and obviously ectoderm will form it, and then the inner gut wall, endoderm is forming it. So I am saying if there is a space in between these two, I'm talking about this space. Okay? I'm talking about this space here. If there is a space in between these two, plus if that space is lined with mesoderm, so we are saying, right, bache? We are saying that coelom is present. That organism is a coelomate, right? Yes. Right? Yes? Tell me. Yes? So see this. So mesoderm is there. So organism is coelomate. Now let's say I'm taking a transverse section of that organism. Now you can visualize body wall, gut wall, there is a space is lined with mesoderm. You can take a transverse section of that. Right? Let's say if, right? Let's say this is the transverse section that we are taking. Okay. So again, this is outer body wall, this is inner gut wall, and I am saying if that is lined with mesoderm, if that space which is present there, if it is lined with mesoderm, then we are saying that organism is what? Then we are saying that organism is a coelomate. Right? Then what do we say? We say the organism is coelomate. Are you getting my point? Then what do we say? We say the organism is a coelomate. Is that clear? Organism is coelomate. Is that clear? Yes, coelom is there. And what will be there in this coelom? The body organs. Right? So if this part is the inner gut wall, this is the outer body wall. Right, bache? And this white colored structure is the coelom here. Organism is coelomate. Now, when we talk about the poriferans, I told you that they are also having a cavity in between. We call it as spongiocele. But bache, here we do not study that thing. Na? We are not saying that there is a proper space in body wall, gut wall, lined with mesoderm or something. No, so a cavity is there. Right? There is an empty space here. But here, there is nothing like this. Right? That's why they are acoelomates. Right? That is why they are acoelomates. Are you getting my point? So just to explain this part. Now see. Now, outer body wall, inner gut wall. Now the space in between, this wala part, this is the lumen part. No. Let's say if somebody is going to take my cross section, outer body wall, right? Then inner gut wall, that empty space in between is lumen. That is right from where the food will pass. So don't get confused. That is how we explain the coelom thing. Right? So what I just said, the word is coelomate. Right? The word is coelom. Yes, the word is coelom. The thing is acoelomate. This thing is pseudo-segmentation. the thing is eucoelomate. That's what we have. So when you talk about the acoelomate without body cavity, so organisms belong to phylum Porifera to Platyhelminthes. They all are acoelomates. They all are acoelomates. Now when you talk about the coelomates, I explained the coelomates as well. So from phylum Annelida to Chordata, all the organisms, they are true coelomates, right? Now here also we will further study Shizocoelomate and Enterocoelomate, but now let me explain you pseudo-segmentations. So when you talk about the pseudo-segmentations, we discuss about the Aschelminthes. Right? We talk about the Aschelminthes. So just have a look here. See, here देखो, true coelom. This is mesoderm having space, right? And now when you say pseudo-segmentations, as I said, you are talking about what? You are talking about Aschelminthes. So बच्चे, here proper cavity is not there, but if you will see the space in between, mesoderm is going to form some pouch-like structure. Mesoderm is not lining this complete surface, complete space, but mesoderm is forming some pouch-like structures. Mesoderm is forming some pouch-like structures here. What mesoderm is doing? Right? What mesoderm is doing? Mesoderm is forming a pouch-like structure. Right, bache? So there is no true coelom. Proper true coelom is not there. Just some pouches are present here. Okay, bache? Pouches are present here. Right? So that's why they are coming under the pseudo-coelomates. So now here, when you talk about the acoelomates in NCERT, see, they are showing this diagram. It is similar to the triploblastic organism. Why? Let's say this is the Platyhelminthes. In their case, mesoderm is there, but in mesoderm we don't see any space, right? We don't see any space. So that's why these are the acoelomates. Is that clear? That's why they are the acoelomates. Is that clear? Now students, when you talk about these eucoelomates, right? So here we have two things. One is Shizocoelomate. Right? And another thing is Enterocoelomate. Shizocoelomate, Enterocoelomate. S for split. Bache, in this type of coelom, in the mesoderm there will be a split. That split will form the cavity. So your Annelida, your Arthropoda, and your mollusca. See, if you can understand it, very good. Otherwise, at least remember the example. They are Shizocoelomates, right? So coelom will formed by the splitting of mesodermal mass. Okay, here coelom will form by the splitting of mesodermal mass. Right? By the splitting of mesodermal mass. Then enterocoelomate, right? So when it is enterocoelomate, here the coelom will form how? By fusion of gut pouches during embryonic stages. Matlab during embryonic stages, from that endoderm thing, some pouches will form, that will fuse, then the coelom will be there. Okay, so if you can visualize it good, I will share an animated video in the Telegram group, in our official Telegram group. You people can check it then. Okay, so coelom will form by the fusion of right, by the fusion of that gut pouches. So here so see, echinodermata, your hemichordata and your chordata, right? Your hemichordata and your chordata are having the enterocoelom, right, bache? So here the diagram is given, but it is just for the coelom, then pseudo-segmentations and acoelomates, right, bache? But here, right, here we have the further eucoelomates. They are further divided, Shizo and Entero. There is a split. And here what is happening? Fusion will form by the coelom will form by the fusion of gut pouches in the embryonic stages. right? So that's what you have to remember, right? That's why even the development, development varies from the organisms which are simple to when we move from one phylum to another, now from simplicity to complexity, we will also see that development will also change. Like here it is Shizocoelom and next is Enterocoelomates. Okay bache, so see this, just see this is endoderm, outer one is ectoderm, and here you have some early mesodermal cell. So when there is a split in that, it will result in your Shizocoelom. When you are talking about the this Enterocoelom, just have a look, this is your endoderm, this is your endoderm. So these gut pouches will arise, some pouch like structure, right? Definitely they are early mesodermal cell. They will arise and slowly see, these pouches like structure will form and they will fuse, right? They will develop, they will fuse, and this is what you are going to get. Okay, this is what you are going to get. Okay, yes, so that's how the coelom will form. So ultimately it is looking like same, but the development, it is changing. Okay, but the way they are developing, it changes. Is that clear? Is that clear? Right? Chaliye. So the next thing is bache circulatory system, that is open and closed, that we will discuss after this segmentation thing. Next thing is the segmentation that we need to discuss. Bache, segmentation also known as metamerism. Segmentation also known as metamerism. Now this is one another character. We will see that some organisms, their body is divided. Matlab, when we define the segmentation, we say that body is divided externally or internally into some segments, externally or internally into some segments, plus there is the repetition of some organs. Ye dekho, body is externally and internally divided into segments with a serial repetition of at least some organs, right? Serial repetition of at least some organs. But when we talk about this segmentation, for the very first time, you will see the true segmentation in Annelida, right? In this phylum, we will see the true segmentation. In which phylum we will see the true segmentation? We will see it in the Annelida. We will see it in the Annelida, the true segmentation. Where do we see this? We will see it in the Annelida. Is that clear? Is that clear? Is that clear? See, so in the Annelida, you can take the example of earthworm. Very clearly, my dear students, very clearly we can see that some external, very clearly these external segments are visible, right? Very clearly these external segments are visible. Plus, internally also segments are there. Just let's say this, externally when you see this, you'll see some grooves. You will see some grooves. What will you see? We'll call them as grooves or annulus. Externally, matlab external segmentation, that is the word that we are using. And internally also the body is dividing. So internally what will we see? This is the septum, this is the septum. Septum is singular, septa is plural, right? Septum is singular, septa is plural. So internally also in the earthworm body you will see that segments are there, right? These segments are there. The septa is divided internally. So these segments are also known as metamers. These segments are also known as metamers. Are you getting my point? Are you getting my point? Right? See this. So externally annulus or groove, externally annulus or groove, internally it is the septum, internally it is the septum, right bache? Internally it is what? It is the septum. These are the metamers, these are the metamers. Are you getting my point? These are the metamers. So this is the septum, this is the septa that we have, right? Right? So that is how body is divided. Now we are saying serial repetition of at least some organs. Just say in earthworm, right? Earthworm is basically belongs to phylum Annelida, nephridia, an excretory organ. We will see the repetition of that as well. Now in our body also, if you take the example of human beings, the vertebra, right? We have the vertebral column. These vertebrae, they are repeated, right? So serial repetition of at least some organs, it is mentioned here now. So that's how we define the segmentation. That's how we define the segmentation, metamerism, right? Metamerism. So we will see the true segmentation in the case of Annelida, and bache, we have the example of pseudo-metamerism or pseudo-segmentation. We also have the example of pseudo-metamerism or pseudo-segmentation, and that example is of a flatworm. Right, that example is of a flatworm, right? Which flatworm? That example is of a flatworm, that is Taenia solium. right? Tinea solium. Okay, so in the case of this tinea solium, what will you see? This is the neck region. This is the neck region. From this neck region, right, we will see some segments will form, and it is not embryonic. They will not form in the embryonic stages, but from this neck region, some segments will form. Just say these are the newly formed segments, and they will be the mature one. We also use the word proglottids for them. Proglottids for them. So here also you will see that body is segmented, but it is not true segmentation because they will not form during embryonic stages. They will not form during embryonic stages. Is that clear? They will not form during embryonic stages. Is that clear? Is that clear? Right? So guys, the way we discuss the incomplete or complete digestive system overall in the animal kingdom, we will see the animals having open circulatory system and having closed circulatory system. So when we are saying open circulatory system, there will be a heart, of course, because it is the pumping organ. So bache, from this heart, जैसे there will be blood vessels, right? Some large blood vessels, they are attached to this heart. Heart will pump the blood, then the blood will, through these large vessels, it will come here in the cavity. Just let's say it's hemocele. So here in the cavity, other body organs are there, right? The other body organs are present in this cavity, and directly, because heart is pumping the blood directly from the large blood vessel, blood is coming. So just imagine I am the heart, right? These are the large blood vessel. When there will be the pumping, blood will come out, and here in this space, body organs, other body organs are placed, they will just swim in that blood. So in the open circulatory system, we do not talk about the blood vessels that are innervating each and every organ and then they are supplying the blood. No, all the body organs, they will just swim there in the blood there. So cells and tissue, they receive directly the blood pumping out of the heart, and in closed one, there will be arteries, veins, capillaries. Just in our case, right, the heart wall, right, it will receive the blood, the oxygenated blood. How? Coronary artery will supply it, to brain carotid artery will supply, right? But here it's not like that. Take the example of cockroach, right? Take the example of cockroach. Having dorsal heart, large blood vessels are connected to this heart, it will pump the blood and blood will come to this space. This space is hemocele. Other body organs are present there, right, bache? So this is the circulatory, open and closed circulatory system. Now, the next thing is again related to the development, that is bache being protostomous or deuterostomous, right? Next thing is also related to the development, and here you are talking about the, right, fate of blastopore. On the basis of fate of blastopore, it is again related to the embryonic development. It is again related to what? Again, it is related to the embryonic development. And here basically we are talking about the fate of blastopore. So in the chapter human reproduction, when we talk about the embryonic development, then we discuss it there. That is blastocyst formation, that pore-like structure formation, everything we discuss there. Now, here we have two words. One is protostomous. One is what? One is protostomes, ya protostomiates. Protostomes, या protostomiates or deuterostomiates. You can also use the word deuterostomes. Yeah, deuterostomous development, protostomous development, whatever it is. So now here we have a trick, and that trick is bad. That trick is bad. That's how I remember it. That trick is bad. So bad means B for blastopore, B for blastopore, A for anus, right? And D for deuterostomes. D for deuterostomes. So basically, when you talk about the organisms which are deuterostomes or deuterostomiates, in their case, just say when the embryo develops, zygote will become multicellular, right? The blastocyst will form a pore-like structure, will also develop. So if that blastopore, if it forms anus, then you will say the organism is deuterostomous. So if blastopore forms the anus first, so right, if blastopore forms the anus, yeah, you can say animals in which anus is formed earlier than the mouth, and the blastopore, it is blastopore forms the anus, and the anus is formed earlier than the mouth. So anus is formed earlier than mouth. So that organisms are what? They are what? Deuterostomes. So again, your echinoderms, so basically echinodermata, your hemichordata, and chordata, they are deuterostomes. They are deuterostomes. Is that clear? They are deuterostomes. Is that clear? Then comes the protostomiates. Here, right, obviously blastopore, because there it is forming anus. So definitely it is forming the mouth here. So mouth will form first and blastopore is going to form the mouth. Right? So from Platyhelminthes to mollusca, they are protostomiates, right? They are protostomiates. Is that clear? They are protostomiates. Is that clear? So see this, protostomiates. So if blastopore form the mouth first, or right, protostomes, deuterostomes may, it will form the, blastopore will form the anus. Is that clear? Is that clear? Okay. Then is the bache notochord. So on the basis of notochord, we divide the organisms. If, see, if it is absent, organisms are non-chordates, animals are non-chordates. If present, organisms are chordates. So notochord, what is it? What is it? It is a mesodermally derived. What is the meaning of being mesodermally derived? That it is made up of mesoderm. It is made up of mesoderm. It's a rod-like flexible structure. Bache, it is on the dorsal side. It is on our dorsal side. Right, bache? So during embryonic development, if this notochord is present, so basically it's a mesodermally derived rod-like flexible structure present on the dorsal side. So if, right, if notochord is present, right, if notochord is present in the life cycle of an organism, it can be in embryonic stage or in adult stage or in both, right? So at least it should be there in their lifespan. It should be there in their lifespan. So if notochord is present, organism will be chordate. If not, the organism will be non-chordate, right? Organism will be non-chordate, right, bache? Right, bache? So these are the basis of classification. These are the basis of classification. Is that clear? These are the basis of classification. Okay. Now, bache, one more thing that you need to remember here is when we talk about the chordates, right? In the case of chordates, it is not just the notochord. Matlab, notochord is definitely there. On the basis of that, we are saying ki bhai, organism is chordate or non-chordate. But we consider four basic characters. We consider four basic characters, right, to keep the organisms under phylum chordata. We will discuss it when we will start your phylum chordata, but I'm writing it here. The first one definitely is the presence of notochord. The first one definitely is the presence of notochord, and the second one is presence of dorsal hollow nerve cord. Right, the second one is the presence of dorsal hollow nerve cord. The third one is the presence of pharyngeal gill slits. The third one is the presence of pharyngeal gill slits. Right, and then there is the presence of post-anal tail. Then there is the presence of post-anal tail. Clear, bache? Then there is the presence of post-anal tail. Is that clear? Is that clear? So these are the four basic characters. On the basis of that, we put the organisms into phylum chordata. So they should have these characters, right? In their, either in their embryonic stage, matlab it can be only in embryonic stage or in embryonic or adult stage, both, right? It should be there. It should be there. These characters, they should be there. Okay. Okay, so just see this diagram. So this structure, this black colored is notochord. Just above this notochord, dorsal hollow nerve cord, pharyngeal gill slits, post-anal tail. So I will discuss it when I'll start the phylum chordata. Okay, so this is all about the basis of classification, and here a flowchart. This flowchart is given, again important because it is in NCERT. So Animalia may ultimately multicellularity is there. So when you are talking about cellular level, matlab subkingdom Parazoa. So poriferans will be there, right? Study of poriferans is parazoology. Then tissue, organ and organ system. So when it is the tissue level of organization, radial symmetry, coelenterata and ctenophora, right? And we know that adult echinoderms are also having the, right? Adult echinoderms are also having the radial symmetry, but their larva will have bilateral. Then bache, organ level plus bilateral symmetry, but body is without coelom. Platyhelminths. Organ system level, bilateral symmetry, false coelom, Aschelminthes, and here this is the thing. So for the echinoderms, you know that adult will have the radial symmetry, larva, adult will have the radial symmetry, larva will have bilateral symmetry. Understood? Understood. So that's what we have in NCERT. So that's it. So next is बच्चे, oh there is a repetition, चलो कोई बात नहीं. So बच्चे next is your phylum Porifera. ठीक है, next is your phylum Porifera. Okay, okay. So before going forward, let's discuss the, right बच्चे, let's revise the name of the phylums again. Let's revise the name of the phylums again. So are you ready? Let's revise the name of the phylums again. Quick. Yes. Let's revise the name of the phylums again. So we have Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and then Chordata. Clear बच्चे? Then Chordata. Done? Clear? चलिए. So let's have a break for 15 minutes. What say? Let's have a break till 8:10 p.m. Okay, then we will continue it further. Okay, then we will continue it further. So just quickly revise the name of all the phylums. Okay, revise it. Done, bache? Revision done? Revision done? Basis of classification clear? Yes, basis of classification clear? I hope now I'm perfectly audible. Yes, I hope now I'm perfectly audible. My voice is not clear. Okay, done. So students, now let's talk about the phylum Porifera, right? What are we going to discuss now? We are going to talk about the phylum Porifera. Yes, we will read the things from NCERT and see, when you talk about the phylums, okay, now because basis of classification is clear to us, right? Right, so you obviously know about that characters, plus you have to remember the peculiar feature, you have to remember the examples here. What you need to focus here? You need to focus on the peculiar feature and you need to remember the examples here. You need to focus on the peculiar feature or you need to remember the examples here. Are you getting my point? Are you getting my point? Yes, right? So let's start this phylum, that is your phylum Porifera. So see, Phylum Porifera, level of organization, cellular, right bache? Symmetry, asymmetrical, but again, mostly asymmetrical they are. Mostly asymmetrical they are. We have the example of the poriferans which are symmetrical, like your Sycon, right? Like Leucosolania, they have radial symmetry, right? They have radial symmetry. So just look at this picture. Look at this picture. Sycon, Stypha, Euspongia, which is also known as the bath sponge, right bache? The bath sponge, and then there is Spongilla. Then there is Spongilla, that is your fresh water sponge. Then there is Spongilla, that is your fresh water sponge, right bache? Fresh water sponge. So they are known as sponges. They are generally marine. So bache, if we are saying generally marine, means some of them are freshwater sponges as well. Like we have the example of yes, like we have the example of Spongilla. Spongilla, do not forget it. It's a fresh water sponge. It's a fresh water sponge, right? They are generally marine, but we have the example of freshwater sponge as well. Mostly asymmetrical, you know that some of them are radial also. Primitive multicellular, having cellular level of organization. Is that clear? Is that clear? Is that clear? So, bache, see, these poriferans, na, let me explain it first, then we will read it from the NCERT. Yes, so these poriferans, now we are saying that they are spore-bearing organisms. What are we saying? We are saying they are pore-bearing organisms. They have pores, they have some openings, right? They have pores, they have some openings. See, this is the diagram, right? This is the picture here. Right, the porifera, look at it, it's quite a detailed picture, right? You can even search it on Google, you will see some structures like this underwater, right? When you look at it, when you look at their body, you will see some minute, small, minute pores are there. We call them ostium. Ostium is for singular, ostia plural, right? Ostium singular. If there is single pore, ostium. If many pores are there, ostia is the word. Their body is perforated. Now it is just having cellular level of organization. It doesn't have a proper circulatory system, proper excretory system or something. They are aquatic as well. So in their case, the water will enter in their body. That water will go out from their body, and during that thing, water is coming in, water is going out, water is coming in, water is going out. That water, right, will provide them the food. The water will also remove the waste material from their body, right? They are that primitive, and as I said, they are animals, but they are attached to the substratum. They are not mobile, They are not movable. They are not movable. Their adults are not movable. They are fixed to a place. So they are fixed to a place. We are using the word sessile. So I am talking about the pores here. I am saying their body is having pores. Minute pores are present here, right? So pore bearing organism. So it is the ostia is the word, right? Ostia is the word. Ostia is the word. These minute pores are present on their body. Right, when you talk about their shapes, they have different shape, cylindrical, vase like, they have different shapes. Now let's see. So these pores are there, these pores are there. So through these pores the water will come inside, the water will come in their body. Now in their case, when you look at their structure, right, when you look at their structure, let's say this is a structure. Now let's say I am studying the layers present here, right? So when you talk about their outermost layer, when you talk about the outermost layer, and body is perforated, pore bearing organisms, they have pores. So when you look at their body now, look at their outer covering, this outermost layer, but this layer is made up of pinacocytes, right? This layer is made up of pinacocytes. This layer, right, the cells that are making it is pinacocytes. So overall this layer is pinacoderm. What is it? It is pinacoderm, and the cells that are making it are pinacocytes. Obviously it's meant for protection. Now here, because pores are there, so here you will see some porocytes. So see this is your ostium through which water will enter, right? Water will enter. Now in their case, the innermost layer that they have, this innermost layer is having some cells, some special cells are present here, right? Some special cells are present here, and that is the thing that you need to remember about these poriferans, right? About these cells, you need to remember about these cells here in their case. जैसे ये देखो. So imagine the structure. I'll show you the other diagrams also. Outermost is pinacoderm, then innermost this layer, but this layer is known as choanoderm. And this layer is made up of special cells. They are choanocytes. I'll come to this. Now in between these two, this space that is present here, in between these two, this is known as, yes, this space present here in between these two, this is basically the mesoglia. What's that? It is actually that mesoglia, or you can call it as mesenchyme or mesohyle, right? It's mesohyle. Sorry, it's mesohyle or mesenchyme it is. It's mesohyle and it's mesenchyme it is. So outermost layer is pinacoderm made up of pinacocyte. The pores here are lined with porocytes. Then this is the choanoderm made up of choanocytes. In between you have mesohyle or mesenchyme. So it is some jelly like part. Here you will see some cells also. Here you are going to see some cells also. Plus here you will see some spicules also. What will you see? You will see some spicules also. Dekho, poriferans, they don't have skeleton like us. So in their case, their body shape and all, how will they get it? That structure, that proper shape, right? So in the mesohyle part, you will see some spicules or some fibers are present. That will give it a shape. Right, and on the basis of that spicules and fibers, this poriferans, they are further classified, right? They are divided into various classes, जैसे calcareous spicule, siliceous spicule, some fibers are there. On the basis of that, we classified further. So when you talk about these phylums, no need to read about classes or order, but in phylum chordata, we have to discuss it up to the classes level. Okay? So it is like that. Right, it is like that. Now next to it there will be the cavity. And that is not the true coelom. I told you already, it is spongiocele. Okay, that is how it is. Now I told you that these cells, they are special cells. You need to remember about them, right? And these type of cells, okay, you need to know about it. जैसे ये देखो, this cell, बच्चे, this cell is your choanocytes. They are choanocytes. They are also known as collar cells. And what is the thing that you need to remember here? It's a flagellated cell. It is having the flagella. So it's a flagellated cell, or we call it as choanocytes, or we call it as collar cells. Right, bachche? So nucleus is there, other organelles are also present. So this cell, right, these are that special flagellated cell. These cells are lining this cavity. Matlab, jaise let's say this is a poriferan, right? This is a poriferan. When you look at the internal structure here, right? That's how you can see. These are the collar cells. We are calling them as choanocytes. See, these are the collar cells. We are calling them as choanocytes. right? They have the outer, they have this outer pinacoderm, right? They have this mesohyl in between, they have this mesohyl in between, and see the space, this part here, what is it? It is the spongiocele. This part here, what is it? It is the spongiocele. This part here, what is it? It is the spongiocele. Are you getting my point? Are you getting my point? So remember, from here, as I said now, their body is having pores, minute pores, ostia. So water will enter through these ostia, right? Ostia means plural, ostium. Then bache, these are the choanocytes, these flagellated cell, collar cells, which we are going to see in the poriferans. Very important it is. These flagellated cells, right, their flagella will maintain the water current. Then that water will enter here in this spongiocele. Then that water will enter here in this spongiocele, and this large single pore, it is known as osculum. It is excurrent pore. The water will go out through this. The water will go out through this. So just look at this setup here. So choanocytes are lining this cavity. We are calling it as spongiocele. We are calling it as spongiocele. These choanocytes are very important because they will maintain the water flow. These choanocytes are very important because they will, right, they will even, like with the water, the food will come now, these flagella will move, so they act like a filter feeder. The food will enter here, the food particles will enter here in these cells, right? So from this part you can remember that in the case of poriferans we will see intracellular digestion. We will see intracellular digestion. Within the cell the food will get digested. Within the cell the food will get digested. And bache, the setup is so interesting here at the base. At the base, this is mesohyl. So you will see here some irregular shapes, some amoebocytes are there, right? So that digested food they will distribute further. They will distribute further. So that's how it is. So their body organization is very, very, very simple. Their body organization is very, very, very simple. Look at this. Look at this. These choanocytes, they are lining these cavity, right? Now, bache, because of this setup, jaise they have the pore through which water will enter. Choanocytes are there, right? The cavity is there. Then the water will go out through osculum. This is what we have. So this is the water canal system. So in their case, what do we see in the case of poriferan? A water canal system is present. A water canal system is present. So these two are very important things here, very important features here. Okay, very important thing and very important features here. Are you getting my point? Yes, are you getting my point? Are you getting my point? Done, bache? Clear? So is it clear? Choanocytes and water canal system. So do not confuse it with the water vascular system. Water vascular system is in echinoderms, water canal system is there. Now the setup that I'm showing you is very, very simple kind of organization. Even water canal systems, right? The body enfoldings are in such a way that here you will further see some other canals also, but that is not the part of our syllabus. So let's keep it very simple. They have a cavity, the spongiocele, right? And through the spongiocele and then through osculum, water will go out. Simple as that. Simple as that. So now please focus on this particular diagram. Ye dekho, outer pinacocyte, pinacoderm is there. These are the choanocytes which are lining the spongiocele, which are lining the spongiocele. Is that clear? Is that clear? So this pathway of water transport is helping in food gathering, respiratory exchange, and the removal of waste. And the removal of waste. Yes? Done? चलिए, so I hope diagrams and everything are clear to you. ठीक है? So what type of digestion? Intracellular. What type of digestion? Intracellular. They are primitive organisms. Here बच्चे, you will also see asexual reproduction as well as sexual reproduction. Right? Both the type of reproduction will be there. Right? So here you are talking about the lower animals which will show both type of reproduction. So look at the example here. Spongilla, freshwater sponge, Euspongia, the bark sponge, and then you have Cycon, also known as Scypha. Okay? So again, even in the plant kingdom, I repeated this part, there is something called BMH. So BMH, B stands for bisexual, B stands for bisexual, M stands for monoecious, H stands for hermaphrodite, H stands for hermaphrodite, H stands for hermaphrodite. Right? B stands for bisexual, M stands for monoecious, H stands for hermaphrodite. B stands for bisexual, M stands for monoecious, H stands for hermaphrodite. That's what you have to remember. If an organism is bisexual, we also use the word monoecious or hermaphrodite for that. So see, Porifera, right? Commonly known as sponges, generally marine and mostly asymmetrical. Generally marine. So when we read this, quickly recall this thing. Spongilla is a freshwater sponge. Right? Quickly revise it with me. Spongilla is a freshwater sponge. Spongilla is a freshwater sponge. Spongilla is a freshwater sponge. Mostly asymmetrical. So again, here, please mark it in your NCERT that we have the sponges that show the radial symmetry. We have Cycon, we have Leucosolania. Right? We have Leucosolania. Is that clear? Is that clear? Now, they are primitive multicellular, done ma'am. They have water transport or canal system, right? Here again, bache, right? You can write it that water, do not confuse it with water vascular system, right? Water canal system and water vascular system is different. It is in the case of echinoderms. Echinodermata are having the water vascular system. Echinodermata are having the water vascular system. So water will enter. Right? Water will enter through minute pores, ostia, in the body wall into the central cavity. What is the word for that central cavity? Spongocoele. From where it will go out through the osculum. So this pathway of water is helpful in how? In food gathering, bache, in respiratory exchange, in the removal of waste, and choanocytes or colloid cells line the spongocoele. Is that clear? So these choanocytes, they line the canal, they line the spongocoele. Is that clear? Is that clear? Is that clear? So digestion is intracellular, intracellular, within the cell. Within the cell, the food will get digested. Within the cell, the food will get digested. Right? Within the cell, the food will get digested. Is that clear? Is that clear? So body is supported by a skeleton which is made up of spicules. body is supported by a skeleton which is made up of spicules. So it can be calcareous, it can be siliceous. It can be calcareous, made up of calcium obviously, siliceous spicule, right? Silica is there. Right, bache? Now spongin fibers are also there, right? Spongin fibers are also there. Okay, so sexes are not separated, जैसे hermaphrodite. So eggs and sperms are produced by the same individual. So means sponges are bisexual. Sponges are bisexual. Sponges are bisexual. So they reproduce asexually by fragmentation. Means a part of their body will get separated and will grow into a new individual. Plus in the sponges, bache, right? We talk about some gemmules, right? We also discuss gemmules. Do you know that? Now the human reproduction in organism chapter is deleted. Now there we used to discuss it. So sometimes they form some spores like structure, right? Some kind of spores like structure, gemmules they are, right? Gemmules they are. Gemmule formation is also there. Then sexually by formation of gametes. Okay, so fertilization here again is internal. What is the meaning of internal fertilization? Means it's not like that they will release their gamete into the surrounding and then fertilization will occur. No, the gametes will form in their body. These amoebocytes actually they turn into different types of cell, right? They will form archaeocytes, right, bache? And that will form the gamete. That will form the gamete, and development is indirect, right? Development is indirect. Are you getting my point? Development is indirect, having a larval stage. So whenever we say the development is indirect, it means zygote is not directly developing into the adult, just in our case, right? Zygote is directly forming an adult. We do not have the larval stages in between. So we have the direct development. But if let's say larval stages are there in between, it will be the indirect development. So that larva is morphologically, means when you look at it, right? When you look at it, you cannot say that it is the larva of adult. They don't resemble morphologically, right, bache? They are different from the adult. Is that clear? They are different from the adult. Is that clear? Right, so remember this. You have the cycon, you have this euspongia, this is the spongilla. This is the spongilla. Okay, ji? Okay, so this is the diagram of the choanocyte. It is a collar cell, right? This is the flagellum. So phagocytosis of food will enter here in the cell, and at its base amoebocytes are there. Digested food will pass here. It will further help in the distribution of that food. Clear, bache? Clear, right? So now let's move to the next phylum. Let's move to the next phylum. That is your Phylum Coelenterata, right? That is your Phylum Cnidaria or Phylum Coelenterata. Okay, let's talk about that phylum. Now again, rest everything we will discuss from the NCERT, but again I will write some important characters here. Students, I am not going to discuss that thing again and again. They are diploblastic, they have tissue level of organization, right, bache? And they are acoelomates. Yes. So I don't think that we need to discuss these things again and again, right? In the basis of classification, we have discussed it already. So now what are the points that we need to keep in our mind? That here you will talk about cnidocytes, right? Cnidocytes. Cnidocytes, nematoblast cells, stinging cells. Yes, bache. So cnidocytes, nematoblast, stinging cells. What do we have? We have the cnidocytes, we have the nematocytes, cnidocytes, nematoblast, stinging cells are there. Right, bache? So these are the special cells that we need to discuss here. Are you getting my point? Yes. So cnidocytes, also known as nematoblast, also known as cnidoblast. Sometimes we also call them as cnidoblast, and they are also known as the stinging cell. One thing, this second is polyp and medusa, right? Here you will talk about polyp and medusa, right, bache? This is what we need to discuss here. Plus, remember about the corals. Do remember about the corals. Okay? Chalo, let me start. Dekho, there is something called alternation of generation. Alternation of generation. जैसे in the case of plants, right? In the case of plants, if you people remember that, right? If you people remember that in the case of plants, right, when we talk about their life cycle, we say na haploid phase is there, diploid phase is there. So in their life cycle, haploid and diploid phase, it alternates. Haploid and diploid phase, it alternates, right? So here in this particular animal, you will see that in their life cycle, sexual stages and asexual stages, they alternate, right? Some sexual stages and asexual stages, they are alternate. This is the alternation of generation, and we also use the word metagenesis for that. We also use the word metagenesis for that. Metagenesis. Haploid, they have sexual stage or asexual stage in their life cycle. Now, just say we are talking about the polyp. Another form is medusa. Right, medusa or medusa, polyp. P for polyp, P for plant. This is my way to remember this. P for polyp, P for plant. It is my way to remember this. So polyp is, bache, just like plant, it is sedentary. It is sedentary. It is not moving here and there. It is fixed. They say it is sessile. Okay, it's sessile. So I hope you know that we have Hydra. Hydra is also the member of this Cnidaria, right? Hydra looks like this. Its structure is like this. Just imagine this structure, right? This structure. I am a polyp. Okay, ji? What am I? I am a polyp. These are my tentacles here. Here I will have mouth. Below the mouth, hypostome. Imagine I'm a hydra. This is the polyp form. Plant-like. This form is plant-like. Matlab sedentary, sessile. Are you getting my point? Sedentary, sessile. Right? Now just imagine, this is polyp, do it like this, go down, do it like this, right? Just bend. This is polyp, do it like this, right? So that form, just let's say here, right? Here, this will be your medusa. So M for medusa, M for mobile, movable, right? Mobile, movable, right? You can take the example of jellyfish here. Jellyfish, Aurelia, it belongs to phylum Cnidaria. Right, it belongs to phylum Cnidaria. So it is inverted. So it looks like umbrella. Yes or no? It looks like umbrella. Yes or no? So here you have the polyp. Here you have the medusa. Here you have the polyp and medusa. These two forms will be there in the life cycle. So on the basis of that, again, it is also classified into different classes. Just as some organisms, they have both. Some organisms, they only have polyps. Some organisms are having medusa. So we have the trick here, that is PAM. What is this PAM? P stands for polyp, A stands for asexually, M stands for medusa. Please do not keep it map. Keep it PAM only. Do not keep it map. Keep it PAM only. Yes, right? It is PAM. So PAM, P for polyp, A for asexually, M for medusa. Okay? M for medusa. Polyp asexually form medusa. Right? Polyp asexually form medusa, so medusa sexually will form polyp. So these two forms, that's why the word is metagenesis or the alternation of generation. Now when you talk about these tentacles, right? Here, right? Obviously layers are there. Just say we talk about the epidermis and all. 1 second. Just say ye dekho. Look at this diagram here. See, this is the mouth part, right? Below the mouth, this is mouth. Below it, hypostome. Okay? These are the tentacles here. What are they? Tentacles here. This is that body. So here also there will be an inner cavity, right? Plus layers are there. Just ye dekho, outer ectoderm, but here you will have mesoglia and endoderm in between, right? And what do you have? You have endoderm in between. Yes or no, guys? Tell me if it is. Yes, please tell me. Is it clear? Please tell me. Is it clear? Yes or no? Is it Clear? Yes or no? Sure? Sure? ठीक है. So they have the layers definitely. They have the layers definitely. So obviously they have that outer epidermis. They have that outer epidermis. Can you see that? This one. Outer epidermis is there. Can you see that? So outer epidermis is having some cells. We are calling them as cnidocyte. We are calling them as cnidocyte. We are calling them as cnidocytes, right? Then in between, as I said, mesoglia, or you can say mesenchyme is there, right? And then innermost layer, which is made up of endoderm. Here you will see nutritive muscular cell. You will see some gland cells like this. Now what you need to understand, so that tentacles, that epidermis is having some kind of cells, right? That epidermis is having some kind of cells here. That this epidermis is having some kind of cells here, right, bache? Right. Yes, some cells are present there. Some cells are present there. So this particular cell is cnidoblast. As I said, cnidocyte. As I said, stinging cells they are. Right, bache? Stinging cells they are. Stinging cells they are. This cell. This cell is present in the epidermis. Now here in this cell, can you see this part? This part here present in between. Bache, this is the stinging capsule. This is the stinging capsule. This is the nematocyst. It's not nematoblast. You will use the word nematocyst. Yeah, right. What is the word here? Nematocyst. So nematoblast is this cell. When you use the word nematoblast, you are talking about this complete cell. But here in between, right, bache? Here in between, when you see this particular structure, this particular structure, it is the stinging capsule and it is the nematocyst or cnidocyst it is. Okay, bache? This capsule-like structure. Can you see that? This capsule-like structure, right, bache? Right, bache? Yes or no? Yes or no? Right? So it is for the offense and defense. This is going to release, right? Matlab this stinging cell, this is undischarged. It is undischarged. It is meant for attachment. It is meant for protection. Yeah, you can say it is meant for offense and defense. It is meant for offense and defense. Right, bache? Right. Yes. Is it clear? So this capsule is having a toxin, hypnotoxin it is. Matlab just whenever they will get their food or they have the prey or they have any predator even for their protection, right? They will discharge this stinging capsule. It is having a thread that will coil around this prey, right? So they will inject the toxin. They will inject the toxin there. Are you getting my point? It is going to inject the toxin there, right? And this toxin, bache, this toxin, right, it causes a kind of nasty sting, right? And it can even paralyze, right? It can even paralyze their particular prey, right, bache? They will release that. So just say your jellyfish, right, that they are also in their tentacles also they are having these cells. So what is going to happen here, right? This is undischarged cell present in the epidermis. So whenever for offense, for defense, right, so they will in that situation they will release it, right? The thread will coil around the prey. So basically then they will inject the toxin. Then it will, so the diagram, so basically the diagram which is given in NCERT, the diagram which is given in NCERT, there is this diagram. So this is showing a discharged cell, discharged nematocyst. Matlab they have used the stinging capsule and their toxin to kill that prey. Are you getting my point? This is discharged. The one that I'm drawing, it is undischarged. Right, it is undischarged, and bache, they can use, right, the coelentrates, they can use the cell once only. Matlab once it is done, once it is discharged, then that cell will be replaced. They can use it once only, right? They can use it once. Are you getting my point? They can use it once. Are you getting my point? Are you getting my point? Are you getting my point? So this one is the discharged one. This one is the discharged one. Are you getting my point? This one is the discharged one. Okay, okay, so they can use it once. They can use it once. So you should know about this cell. Moreover, in the phylum, again, a very important thing, right? Again, a very important thing, a very, very, very important thing. In Phylum Cnidaria, for the first time, you will see the network of nerve cell, a diffused network of nerve cell. You can say a primitive network of nerve cells will be there, right? A primitive network of nerve cell is present. This is the first phylum in which we will see that primitive network of nerve cell. Okay, okay, but in sponges, in poriferans, it is absent, right? In poriferans it is absent. Now the point is that if they do not have the nerve cells, then how are they going to respond? How are they going to revert? Right? How are they going to respond? How are they going to revert how are they going to respond to the stimuli? So in the case of sponges, bache, right, obviously let's say that chemical attraction thing is there, but in their body you will not see the network of nerve cells, right? Even if it is primitive, you are not going to see the network of nerve cells there. Okay, bache? Done? Okay? Note down these points. Okay, chaliye. So that's it, that's all. Okay, that's it, that's all. So this is all about the main characters of coelenterata. So yes, this is for the first time in this phylum we will see the diffuse network of nerve cell. In the sponges they don't have that, but yes, they respond to the stimuli. Chemicals are there, right? Chemicals they will release, but otherwise they don't have that diffuse network of nerve cells. So they are aquatic, mostly marine, right? They are aquatic, mostly marine. Yes or no? They are aquatic, they are mostly marine. Okay, okay, sessile or free living, radially symmetrical. Okay, bache. So Cnidaria is the word which contain the stinging capsule or nematocyst, right? So they are present on the tentacles and the body. They are used for encourage, means attachment, defense, and for capturing of prey. So they exhibit tissue level of organization and diploblastic. Simple, it is already discussed. They have a central cavity, that is gastrovascular cavity. Now wait, yes, look at this diagram. Look at this diagram and this diagram. This cavity we are talking about. Okay, this cavity we are talking about. Clear, bache? This cavity we are talking about. So this is the paragastric cavity. This is the paragastric cavity, right, bache? Or you can say that, see, in their case, in the cases of your cnidarians, even in the cells the digestion will be there, but here in this cavity also they will digest the food. So here it will be intracellular and extracellular digestion. What type of digestion we are going to see here? Intracellular and extracellular digestion will be there. Intracellular and extracellular digestion will be there. Clear, bache? Clear, bache? So look at this part. Look at this part. Look at this part. See this. This is the undischarged cell. This is the discharged cell. This is the undischarged cell. This is the discharged cell. Is that clear? Is that clear? Okay. So we have tentacles, mouth is there. So you have epitheliomuscular cells, right? This here. Look at this. you have epitheliomuscular cell, you have cnidocytes, right bache, and here see muscle fibers are also present, sensory cells are there. So this is about the cavity, gastrovascular cavity, a paragastric cavity it is, right bache. So here also the food digestion will occur. Here also the food digestion will occur. Okay? Right? So polyforms which are stationary, medusa which is free floating, mouth is towards the downside. Then I'll show you the examples. Okay? Okay. So they have central gastrovascular cavity with a single opening, mouth on hypostome. Okay? So digestion is extracellular and intracellular. So in poriferans it was only intracellular, but here extra and intra. Some of the cnidarians, they have corals. Example, corals have a skeleton composed of calcium carbonate, right? So corals, right? So corals, some of cnidarians, just corals, their skeleton is made up of calcium carbonate. So they exhibit two basic forms, polyp and medusa. So I told you, polyp is sessile, matlab attached to the substratum. It is cylindrical, just like your hydra, just like your adamsia. Adamsia, you know na? Sea anemone. Sea anemone. So sea anemone is fixed over a empty shell. That shell will have the hermit crab. So with the help of that crab will take the shell here and there, sea anemone will move, and that sea anemone, its tentacles, its cells are going to protect that crab. So that type of association we see in their case. Okay, so medusa is umbrella shaped, free swimming, just like your jellyfish. So cnidarians which exist in both forms, they exhibit alternation of generation. Clear bache? So we have Physalia, Portuguese man of war, right? Actually, how do they attack? Matlab they'll just hide, and whenever they'll see their see the one where they want to attack, they just attack. Physalia, they don't confront their enemy, they just attack. But how? They will just hide, right? They will just hide. They are not just going to confront their enemy and then suddenly they will attack. So it is believed that even Portuguese people, that's how they used to attack. So that's why Physalia is considered, Physalia is called as Portuguese man of war. Okay? So Physalia is also showing both, right? It is also polymorphic. It is also polymorphic. It is also having both the form. Right, Physalia. So look at this. This is the nematophore cell, cluster of polyps. This venomous tentacles they have. Okay. Then Adamsia, your sea anemone. Penatula, you should know the common name. Sea pen, Gorgonia, sea fan, and Meandrina, brain corals. Meandrina, brain corals. Is that clear? So these are the diagrams. Your Aurelia, medusa form. Right. This is sea anemone. This is the polyp form. Okay. This is the polyp form. Okay, done. Chaliye, next is Ctenophora. Next is Ctenophora. So write down the basic characters. Write down the basic characters of the Ctenophora in the chat section. Right, the basic characters. Write down. Okay. Hello. Good. So Phylum Ctenophora, again, tissue level of organization. Tissue level of organization. Diploblastic they are, right bache? And one thing that you need to remember, tissue level, diploblastic, and again they are also acoelomates. So symmetry will be radial here. Okay, as per NCERT, as per NCERT. Now, when you talk about the Ctenophora, the one thing that you need to remember is exclusively marine. One thing that you need to remember is exclusively marine. They are exclusively marine. Right, bache? What are they? They are exclusively marine. Okay? Okay? So exclusively marine means they will only be there in the marine water. So basically there are 3 phylums. I'm not talking about the subphylums. I'm talking about the phylums where organisms are exclusively marine. Your Ctenophora, your Echinodermata, your Ctenophora, Echinodermata and Hemichordata. Your Ctenophora, your Echinodermata and your Hemichordata. Here they are exclusively marine. What are they? The organisms are exclusively marine. What are they? They are exclusively marine. Okay? Okay? Exclusively marine they are. Yes, they are exclusively marine. Okay, so you have phylum Ctenophora, you have phylum Echinodermata, you have phylum Hemichordata. These 3 subphylum where organisms are exclusively marine. This is the one thing that you need to remember here in Ctenophora. Secondly, here we will see only sexual reproduction will be there. Just even in your this Cnidaria, you will see budding will be there in Hydra. Budding will be there in Hydra, but here you will only see sexual reproduction. Here you will only see sexual reproduction. Here you will only see sexual reproduction. What will you see? Sexual reproduction. So tissue level of organization will be there. Diploblastic organisms, acoelomates, radial symmetry. Organisms are exclusively marine. Organisms are exclusively marine. Is that clear? Organisms are exclusively marine. Is that clear? Is that clear? Right, bache? Then, now when you talk about the special characters or something, here we will see lasso cells. Lasso cells. They are adhesive cells. They are also known as coloblast cells. They are also known as coloblast cells. Coloblast cells. Right? Lasso cells are there. They are adhesive in nature. Adhesive in nature. And here in this phylum, you will see bioluminescence. What will you see? You will see bioluminescence. The organisms, they emit light. They emit light. We will discuss about it. First of all, in this particular phylum, what you need to remember? What you need to remember? This. What you need to remember? This. You need to remember the tentacles. You need to remember the tentacles. Right? Here you will see the tentacles are there. It's not mandatory that all the organisms will have tentacles. No. One thing that you need to remember here is that some organisms, they will have tentacles. Some organisms, they lack tentacles. So, on the basis of this, their presence or absence, we further classify them. But they are not using these tentacles for their movement or something. No, they are not using it. When you look at their structure, right? When you look at their structure, right? You will see that they will have comb plates. Just we call them as comb plates also, right? They have different names. I'll come to this. On their body, you will see cilia is present. Cilia is present like this. Matlab cilia is present in their body. Just ye dekho. This is the structure that we have here. This is the structure that we have here. See this. Cilia is present there. Cilia is present there. So these eight rows of cilia. These 8 rows of cilia, they are present here. 8 rows. 1, 2, 3, 4, 5, 6, 7, 8. This will help in the movement. So usually we see cilia in the lower organisms, just in the unicellular organisms. So they are multicellular, but still in their case, it is the cilia that will help in the movement. It is the cilia that will help in the movement, right? So body is having 8 external rows of ciliated complates which help in locomotion. So these organisms, right, ctenophores, they emit light. They have this property. Living organisms are emitting light. So that's why we are using the word. That's why we are using the word. Which word are we using? We are using the word bioluminescence. Right, bache? Right, bache? So bioluminescence. Okay? So here you will see nematoblast cells. They are absent. Nematoblast cells are absent here except one species. Matlab here only one species is having in the ctenophora, there is one species that will contain these nematoblast cells. Otherwise all, right, in all other phylums, in all other organisms, it is not present there. Right, bache? So they have lesser coloblast cells which are adhesive, which help in capturing the prey. Right, bache? Which help in capturing the prey. Is that clear? Which help in capturing the prey. Is that clear? Is that clear? So there they will only show the sexual reproduction, right, bache? And these cells are present there. So look at the diagram. Look at the diagrams here, right? Ctenoplana, pleurobrachia. Okay, ctenoplana, pleurobrachia. Is that clear? Sure. Is that clear? Is that clear? Okay. Okay. So comb jellies, sea walnuts, right? They are known. These are the common names there. Okay. So your yes. So just say pleurobrachia example is there. Pleurobrachia. It is also known as sea gooseberries. Sea gooseberries they are. Okay. So let's quickly recall it from the NCERT. So see, sea walnuts or comb jellies. Okay, they are sea walnuts or comb jellies. These are their common names. So sea walnuts, that example is not given there. Okay, that example is actually Hormifora, right? Hormifora. That is actually a ctenophore which is known as sea walnut. Exclusively marine. Exclusively marine, bache. Radially symmetrical, diploblastic. Okay, so they have 8 ciliated rows. This point is important. Okay, digestion here again, both extracellular, intracellular, and bioluminescence is there. So reproduction will take place only by sexual means. Important it is. So sexes are not separate again. They are also monoecious. Okay, they are also monoecious. Fine. So fertilization is external with indirect development. So examples are given here. Okay. Any doubt? Any doubt? Any doubt? Yes? Done? Next phylum is Platyhelminthes. Here you are talking about the flatworms. Here we are talking about the flatworms. What are we discussing here, students? We are talking about the flatworms here. So what do you know about it? Yes? Guys, what do you know about it? Bache, excretory organs are not well developed in the lower organisms. If in lower organisms you are thinking you will see proper excretory organs, no. Just in platyhelminths, right? One student is asking a question, I'm answering that. Just platyhelminths mein you will talk about it. In platyhelminths we will discuss protonephridia. In platyhelminths we will discuss flame cells, right? We will discuss flame cells. In this platyhelminths, we will talk about the flame cells, we will talk about the protonephridia, we will talk about the solenocytes. They are actually, right, bache, they are actually what? They are the excretory organ. Right? They are the excretory organ. This cell will help in the excretion or the osmoregulation. But in that, sponges, I told you, water canal system will help there. So there you are not going to discuss that. Are you getting my point? There you are not going to discuss that. Okay? So do you have any other doubt? Do you have any other doubt here? Or can we just start this phylum Platyhelminths? Do we have any doubt here? Or can we just start this phylum Platyhelminths? Yes. ठीक है बच्चे मतलब साई are you actually attending the lecture or not? I think I have explained it already. So what is the next phylum? Next phylum is phylum Platyhelminths. ठीक है here बच्चे organ level of organization will be there. Organs will form. Organ level of organization will be there. Organ level of organization will be there here. What will be there? Organ level of organization. Is that clear? Is that clear? Tell me. Is that clear? Yes or no? Sure? Sure. So now they are acoelomates. They don't have coelom, but remember their symmetry is bilateral. Their symmetry is bilateral and they are triploblastic. Their symmetry is bilateral and they are triploblastic. Their symmetry is bilateral and they are diploblastic. Right, bache? So why do we say that they are? Why do we say that they are flatworms? Because of their structure. Because of their structure, it is dorsoventrally flat. Matlab consider a leaf. Some structures like that. Their body is just like that. Okay, and here you will see that mostly they are endoparasites. I'm not saying all of them are endoparasites. Mostly they are endoparasites. So on the basis of this thing, right, whether they are endo, ecto, ya fir free living, we further classify them. We further classify them into different classes. Okay, so see, mostly endoparasite found in animals, including human beings, and they are bilaterally symmetrical. They are bilaterally symmetrical. So now let's discuss about it. Okay, bache? Let's discuss about it. Okay, so when you talk about these tapeworms, okay, when you talk about these tapeworms, other than their general characters, we know about their general characters, right? We know about their general characters, right? So they are basically the worms, right? They are basically the worms. They are endoparasites. So when we are using the word endoparasite, again, I'll discuss with the discuss the most detailed, right? I will discuss the most what do we say? Most important characters here, then we will read it from NCERT. Endoparasite, it is living within my body. It is living within my body. So if it is living within my body, its body wall, its outer covering should be like that. Its outer covering should protect it from my enzyme. I have an immune system also. If a worm is living within my body, so obviously they need to protect itself from it needs to protect itself from what? From my enzyme. Plus, they need to get attached to my cells also. Right, bache? Now, because they are in my body, I am eating something, they are going to feed on that food. They don't have you will see if they will have proper digestive system or something. No, you are talking about the flatworms, and here we are mainly going to see blind sac body plan. What will we see? Blind sac body plan. Here, right, those who are endoparasite, in their case even locomotory organelles are not there, right? In their case you will see some parasitic adaptation, jaise ki they will have hooks and suckers. So with the help of that hooks and suckers, they will attach to the host's body, they will attach to the host cell, right? From their body itself, outer covering, they will absorb the food directly. They will absorb the food directly. Okay, bache, they will absorb the food directly. Okay. Okay, they will absorb the food directly. Okay, bache. So what are we talking? We are talking about the endoparasites, right? So we are saying they are endoparasites. So they have hooks and suckers which will help them to attach to the cells of the host, right, bache? So these things are there in their parasitic form. These things are present in their parasitic form. Now I say, as I said, if they are parasitic, they are going to live within my body. They need to protect itself from what? They need to protect itself from what? They need to protect itself from the host's digestive enzymes. Okay, bache? So here you will see that when you talk about their outer body wall, when you talk about their outer body wall, right, bache, outer covering, their epidermis, right? So there, here you will see that their body wall is having a thick cuticle. Their outer body wall, it is covered with a thick cuticle. It is having what? It is having a thick cuticle. Are you getting my point? It is having a thick cuticle. Are you getting my point? And for that, we also use the word tegument. We also use the word tegument. Just here, epidermis will be there. It will be ciliated, right? Plus thick cuticle is covering it. That is known as tegument, right? And that will protect the parasite from the, right? That will protect the parasite from the host's digestive enzymes. That is going to protect the parasite from the host's digestive enzyme. Is that clear? Is that clear? They have the tegument. Okay, bache. Okay, bache. So acoelomates they are, right? Yes, right? Acoelomates they are, we know that. Acoelomates, but they are triploblastic. So when you talk about their body, na, they say in their body, at places we will see some loose mesodermal, right? Some loose mesodermal tissue is there. A loose mesodermal tissue, some loose mesodermal cells or tissue are present here, right, bache? Right, in their body. Okay, so that is known as mesenchyme or parenchyma. That is known as mesenchyme or parenchyma. That is known as mesenchyme or parenchyma. Clear, bache? Or parenchyma. Is that clear? Is that clear? Yes. So again I am telling you, they have blind sac body plan. They have blind sac body plan. Yes or no? They have blind sac body plan. So it means their digestive system will be incomplete. Their digestive system will be incomplete. Yes or no? What kind of digestive system do they have? They will have incomplete digestive system. They will have incomplete digestive system. Is that clear? Right. Now one thing, they said tapeworm. There is an example which is given in NCERT, that is of tapeworm. That is your Taenia solium. Right, Taenia solium. It is an endoparasite. It is an endoparasite. It lacks digestive system, bache. Why? Because it is a endoparasite. Right, it will get that digested food. It will directly absorb it from its outer body. Okay, right. So it is the tapeworm. It is the Taenia solium. It is the tapeworm. It is the Taenia solium. It is the tapeworm. It is the Taenia solium. Right, bache? Right, bache? Right, bache? Yes. Okay. Clear? Clear? Right? It lacks the digestive system. Now, here. Okay. Baki, that respiratory system, no. Directly through the body covering, they will exchange the gases. Right. Now, in their case, we will see flame cells. Right, why do we call it as flame cells? Because of its structure. That flame cell is also known as protonephridia, right? That is also known as solenocytes. That are also known as solenocytes. So they have protonephridia, flame cells, protonephridia, solenocytes, bache. So it is meant for excretion. It is going to help in excretion. Very important it is. And osmoregulation as well. So what is osmoregulation? right? Maintaining salt and water balance in the body, right? Maintaining salt and water balance in the body, maintaining salt and water balance in the body. Is that clear? Is that clear? Sure, sure. Sure. ठीक है. So excretion and osmoregulation. Clear? In their case, what will we see? Right? Done? So, flame cells, protonephridia, solenocytes. So, excretion and osmoregulation is there. Clear, bache? It is going to do that. Okay? Clear? It is going to do that. Okay? Done? So, now, bache, when you talk about the nervous system here, right? When you talk about the nervous system here, yes, we will see the nervous system, right? There will be a nervous system in their case, and here we will see ganglia. What are we going to see? We are going to see ganglia. What are we going to see? We are going to see ganglia here, right? We are going to see ganglia here. This ganglia is composed of nerve cells. This ganglia is composed of nerve cells, right? Ganglia. Ganglia means composed of cell body. Just we have the neurons now. Neurons, their cell bodies, right? Their cyton, their cell bodies, they will aggregate, right? They are forming the ganglia. So when you talk about the nervous system, it is ladder-like, right? It is ladder-like. What kind of nervous system is there? It is ladder-like. Just imagine something like that. Longitudinal nerve cords will be there, right? Transverse nerve cords will be there. So it will be ladder-like, okay? It will be ladder-like. It will be having a nerve ring and longitudinal nerve cords, right? It consists nerve rings, okay, and longitudinal. It consists nerve rings and longitudinal nerve cords, right? Clear, bache? Is that clear? Sure? Are you sure? Are you sure? Done? Any doubt here? Yes, any doubt here? So brain is there, longitudinal nerve cords are there, and obviously transverse commissures are there in between. Okay, excretory system, I told you protonephridia will be there, and sexes are mostly united. Okay, here also you will see sexes are mostly united. Now I was about to explain one point, I just one more point is there, I just forgot to explain. What was that? अच्छा चलो छोड़ो. प्रोटोनेफ्रिडिया फ्लेम सेल्स इनकम्प्लीट. ठीक है, देखो इन देयर केस जैसे प्लैटीहेल्मिंथ्स में आई टोल्ड यू मोस्टली एंडोपैरासाइट. सो वी नीड टू रिमेंबर द एग्जांपल ऑफ फ्री लिविंग प्लैटीहेल्मिंथ्स. दैट इज योर डिगेजिया. दैट इज योर डिगेजिया एंड दिस इज प्लेनेरिया. दैट इज डिगेजिया एंड दिस इज प्लेनेरिया. This is digesia and that is planaria. Is that clear? Right? And it is having the great power of regeneration. Here we will see regeneration as well. Just say, I don't know whether you remember that diagram or not. Something like that. Something like that. Do you remember that? So it shows a great power of regeneration. Right? Digesia is free living. Now here also sexes are united, matlab they are monoecious. But one thing bache I would like to mention here, see after this na, after this Platyhelminthes, we have the next phylum, that is Aschelminthes. After this Platyhelminthes, we have the next phylum, that is Aschelminthes. So when you talk about the Aschelminthes, in the case of Aschelminthes, we say that for the first time we see the sexual dimorphism in the phylum Aschelminthes. I hope you know about it, that in the phylum Aschelminthes, for the very first time we are going to see the sexual dimorphism, right? But here, bache, what's the point? In the Platyhelminthes, the diagram which are given, one is Tinea solium, that is tapeworm, right? This is those who eat pork. If that pork is having this if it is infected, if in the muscles of that pork, right, cyst is there, cyst of tinea, then if a person is going to eat it and if that pork is undercooked, that person will get the infection, right? That person will get the infection, teneases from this, right? Teneases. So here human is also the host and pig is also the host. Then it is the Fasciola hepatica, right? This is the Fasciola hepatica, right? Again, it is also a parasite, not of humans, of course. So this Digasia is also the example, also known as Planaria. There is one more example, bache, that is Schistosoma. That is also known as blood fluke, right? There is one more parasite here that is known as Schistosoma. That is also known as blood fluke, right, bache? So it is there in the veins of, right? It is in our case, Schistosoma, blood fluke. So it is found in the veins of human bladder. It is found in the veins of human bladder and intestine. Bache, here you will see that here the sexes are separate. Why am I telling you this? Because here the sexes are separate. Why? Because here the sexes are separate. We are talking about the schistosoma, that is blood fluke. So veins of human bladder and intestine, right, bache? So it is found there. So it is unisexual, where male carries the female. Here it is unisexual. Male carries the female. Yes, male carries the female. Female how? Yes, bache, in the groove, okay? In a groove-like structure. That is known as gynecophoric canal. That is known as gynecophoric canal. So in that gynecophoric canal, we'll see, male on the ventral side, it will carry the female, okay? Okay, it will carry the female. Is that clear? In a groove, gynecophoric canal. Right. So here you will see the sexual dimorphism is there. No doubt, properly in the phylum Aschelminthes you will see. Right. In phylum Aschelminthes we will see sexes are separate. But it's not, and we say that this is the first phylum of the animal kingdom where we will see sexes are separate. But here in the phylum Platyhelminthes, this is the exception that we have. Okay, this is the exception that we have. So this particular schistosoma, it is also having two hosts to complete its life cycle. So if any parasite is having two hosts, digenetic. If single host will be there, monogenetic. And here primary host is human, right? Primary host is man, right? Primary host is man. So will you please tell me what is the meaning of being a primary host or a secondary host? Yes, what is the meaning of being a primary host or the secondary host? Yes, what is the meaning of being a primary host or the secondary host? Primary host, right? The one in which the sexual stage of that parasite will be there, right? The organism in which the sexual stages of parasite will be there. Primary host. Just in the malaria, right? In the case of malaria, we have the plasmodium. Plasmodium, it causes the malaria. It is the malarial parasite. So in the case of malaria, human is the secondary host. Female Anopheles mosquito is the primary host because sexual stages of that parasite are in the female Anopheles, right? So primary host is human here. Secondary host is garden snail here. Secondary host is garden snail here, right? Secondary host is garden snail here. Clear, bache? Clear, bache? Is that clear? Is that clear? Right? So schistosoma example, and I told you about that ladder-like nervous system now. So this is your Digasia, ladder-like nervous system. Here you have these cerebral ganglias, right? These are the transverse commissures. These are the longitudinal nerves. Okay, and this is the flame cell, also known as protonephridia, also known as selenocyte. Okay, also known as Protonephridia also known as selenocyte. Is that clear? Is that clear? Yes. Pakka? Is that clear? Sure? Okay. Done. So see, this is the Taenia solium, tapeworm, and here what will you see? You will see pseudo-metamerism. You will see pseudo-metamerism here. So pseudo-metamerism, false segmentation. Why? Why it is the false segmentation? Because from the neck, right? Because from this neck region here, the segments, these proglottids are formed. So here you will see the mature one, here you will see the newly formed one. So this is the liver fluke. Right, what's that? It is the liver fluke. Okay, okay. So liver fluke may be bache here. Again, it is also parasitic. Okay, again it is also parasitic. Right. So it is found in the bile duct of the liver of sheep and goat. Okay, in the bile duct and liver of the sheep and goat. Okay. Okay, so here also, right, the other host is snail. Here also the other host is snail. Okay, chalo, done. Next is Phylum Aschelminthes. Next is Phylum Aschelminthes. Right, Phylum Aschelminthes. Okay, chaliye. So bache, phylum is the Aschelminthes. You are talking about the roundworms here. What are we talking? We are talking about the roundworms here. Why? Because when we take the cross section, now when we take their cross section, we will see that circular outline. So we are calling it as the roundworms, right, bache? We are calling it as the roundworms. Okay, Phylum Aschelminthes. Here, organ system level of organization. Yes, organ system level of organization. Triploblastic, Pseudocoelomates, bilateral symmetry. Okay, that's something that you already know. Other than that, here you will see the muscular pharynx. Here, what will you see? You will see muscular pharynx. What will you see? Muscular pharynx. Okay, okay. Right, what will you see? You will see muscular pharynx here, number one. For the excretion, we will see H-shaped rennet cells are there. And that's not the only thing we have. We have other cells also. H-shaped rennet cells are there for the excretion. Plus here you will see sexual dimorphism. Sexual dimorphism will be there. They also infect plants. This is also known as Nemathelminths. Yeah, nematodes belong to it. Nematodes belong to it. So here muscular pharynx, H-shaped rennet cells, and the sexual dimorphism. Okay, and the sexual dimorphism, that's what we see. Other than that, we will have some sense organ like papillae, amphid, phasmids, right? Sense organs like papillae, amphids, phasmids are present. Right, they are present there. Is that clear? That's what you are going to see. Okay, that is what you are going to see. Clear, bache? Okay. Okay. So chalo, let's discuss it. right? Let's discuss it. The body of the Aschelminthe is circular in cross section. That's why the name is roundworms. They may be free living, not infecting anyone, living alone, aquatic and terrestrial, or parasitic in plants and animals. Can you quote some example? Yes, any example that you guys can quote? Any example that you guys can quote? Yes, they say one is one example is this. In the case of plants, there are many. I'm picking up this example. It is in the biotechnology part as well. Biotechnology applications, do you remember? In the biotechnology, in the application parts, we have this particular nematode. It infects the tobacco plant. Yes, bache, it infects the tobacco plant. So it is Melodygen incognita. So bache, it is going to affect the roots of the tobacco plant, and thereby it reduces the yield. So if you remember the RNA interference technique, right? The RNA interference technique, if you remember, RNA interference technique is used to form the transgenic tobacco plant, right? To form the transgenic tobacco plant. ठीक है बच्चे? ठीक है बच्चे? So this is in the case of plants. This example is given in your NCERT only. ठीक है? So in the case of animals, we will talk about we have many examples. We will talk about the Ascaris. ठीक है? Wicheria vencrofti is there, Wicheria malai is there, right? And Cyclostoma. Many examples are there. So now come to this. So roundworms, they have organ system level of organization, bilaterally symmetrical, triploblastic, pseudo-segmentation. Done. Well developed. Yes. Their alimentary canal is complete. So if I am saying the alimentary canal is complete, so can I say that tube within tube body plan is there? Can I say that tube within tube body plan is there? Yes or no? Can I say that they have tube within tube body plan? Tube within tube body plan, because the digestive system is what? It is complete. Okay. Now what is the next part here? An excretory tube removes the body waste from the body cavity through the excretory pore. Dekho. Here we have the diagram. Just see this. Look at this. Okay. So here this is of Aschelminthes, right? But so as I said, they have H-shaped rennet cells now. H-shaped rennet cell. This H-shaped structure they have. Right? They have this X-shaped structure. Just excretory pore. These canals are there. Right, bache? Right. So H-shaped. Right? What do they have? They have H-shaped, and it is having the excretory canal which remove the body waste from the body cavity. Right, bache? Okay, so look at this. So that's what you have to remember. H-shaped rennet cells are there. Okay, H-shaped rennet cells are there. And I told you about the sense organs now. Jaise papillae, amphids, phasmids are there. Right, papillae, amphids, phasmids are there. So these two are the chemoreceptors. Right, these are the chemoreceptors. And this one is the tango receptor. So please tell me, what is the meaning of tango receptor? Yes, please. Please tell me, what is the meaning of tango receptor? Quick. What is the meaning of tango receptor? Yes, tango receptor. Bache, they can detect, they can sense touch and pressure. Right? Chemoreceptor, that will sense chemical. Right? Chemoreceptor, that will sense chemical. Clear, bache? It will sense chemical. Okay. Clear? Sure. So they are pseudo-coelomates. They are pseudo-coelomates. Okay. They are pseudo-coelomates. Okay. So even, but see, as I said, they are pseudo-coelomates. So in this pseudo-coelom, no, in this pseudo-coelom, we will see a fluid, and that fluid will maintain the pressure there. Okay, we will see the fluid, and that fluid is going to maintain the pressure. That fluid is going to maintain the pressure there. Fluid is present, that fluid will maintain the pressure there. Fluid is present, that fluid will maintain the pressure. So here they don't have the proper skeleton, but because of this fluid, you can say it will give it a shape. So we can say a kind of hydroskeleton is present here, right? A kind of hydroskeleton is present here because it will maintain the body shape. right? It will maintain the body shape. Okay, done? Sure? Yes, is it clear? Okay, Baki, see, I already told you that here we will see the sexual dimorphism. We will see the male is shorter than female, right? The body of the male will be curved here, right? So that's it. That's what you need to know. So Baki, dekho, sexes are separate, they are dioecious. Males and females are different, distinct. Often females are longer than males. Fertilization is internal and development may be direct, right? May be direct or indirect. The development may be direct or indirect. जैसे ये देखो, here ascaris is given बच्चे. Ascaris, it's a roundworm. This ascaris, it causes ascariasis, right? It causes ascariasis. And बच्चे, it is monogenetic, means it is having the single host only. It is having the single host only. It takes a 10-day tours in our body. ठीक है? Then is which area? Next is which area? बच्चे, that which area is the... It is also known as filarial worm. It is also known as filarial worm. It is बच्चे viviparous, right? जैसे we have oviparous means eggling, right? Then viviparous, that is going to give birth to the young ones. But here you are not going to see any placental connection or something, right? Here you are not going to see any placental connection or something. ठीक है बच्चे? You are not going to see any placental connection or something. So जैसे we have which area? Vencrofty. So it causes elephantiasis, right? This worm causes elephantiasis or filariasis, right? Or filariasis, elephantiasis or filariasis. So your culex, right? Culex, the carrier host here, female culex. So culex, carrier means the vector. It will spread it, this infection. So female culex mosquito will spread it. Okay, so elephantiasis, so it can block the lymph nodes and the swelling will occur. You can check the pictures, right? You can check the pictures of it. So elephantiasis, so this is important. Which area? Okay, so it is the filarial worm. It is viviparous, right? It will directly form that larva. Sorry, it is basically the development here will be what? The development here will be what? The direct development will be there. ठीक है बच्चे? Okay. Okay, so it is also digenetic because it is having two hosts. Okay, it is also digenetic. Why? Because it is having two hosts. Okay, done. So female will deliver microfilariae there, right? Female will deliver microfilariae. Female will deliver microfilariae. Okay, बच्चे? Okay. These are the juveniles. Done. That's why it is the viviparous. That's why it is viviparous. Okay, and it is not the only viviparous worm. There are others also. Do check it. Do check it. Here they go. So Encyclostoma, it is the hookworm. So again, this is your homework. You will tell me in the comment section that why do we call it as a hookworm? Okay, why do we call it as a hookworm? Okay. Okay. So this is the male member here and this is the female member here. Okay. So as I said, it is the which area which is viviparous. Other than that, there is one more example, Trichinella. That is also viviparous. Chaliye. So next is Phylum Annelida, right? Next is Phylum Annelida, where for the first time you will see true segmentation, that is metamerism, or they are having coelomates, obviously. Coelomates, these are the characters which we are going to see here in this particular phylum for the first time. So we are done with the 5 phylums. Porifera done, Cnidaria done, Ctenophora done, Platyhelminthes done, Aschelminthes done. Now we will talk about Annelida. So let's have a quick revision. So see, we started from the basis of classification, done. Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes. Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes. Done. Chalo. Yes, Sai, the statement is correct. Okay. So they may be aquatic, freshwater or terrestrial, free living, and sometimes parasitic. They exhibit organ system level, bilateral symmetry, triploblast, and they are coelomates. What are they? They are coelomates. Now, what's the point that you need to keep in your mind here? We have 3 examples here. Okay, I'll discuss everything. We have 3 examples here. We have the example of Nereis, which is also known as rugworm. We have your earthworm, Pheretima species, of course, and we have Hirudinaria, that is your leech. So Pheretima, that is your earthworm. So students, these 3 examples are given in NCERT. These 2 are again monoecious. Matlab, if in Aschelminthes for the first time we have seen the sexual dimorphism, that doesn't mean it will last for all the phylums. No, again in Phylum Annelida you will see these 2 are monoecious, though they prefer, just earthworm prefers cross fertilization, but it can still go for the self fertilization. So Nereis is dioecious. So these are the 3 examples which are given in NCERT here. Yes, these are the 3 examples which are given in NCERT here. So Nereis is there, this Pheretima is there, Hirudinaria is there. Firstly repeat it, then we will discuss it further. Yes. And again, before going forward, here bache, even in the Aschelminthes, I just forgot to mention it, even in the Aschelminthes, whenever you talk about the body wall, na, right, obviously outer cuticle will be there, epidermis and muscular layer is present even in the Aschelminthes. Okay? Outer cuticle, epidermis and muscular layer is present there. Okay? So outer cuticle, then epidermis and muscular layer is present there. So cuticle, obviously non-living, will protect this organism from host enzyme. Epidermis is multinucleated, it is syncytial. And when you talk about the muscular layers, you will only see, yes, what will you see? You will only see the longitudinal muscle fibers. Okay, so now we are discussing the Annelida, and I'm telling you to remember these 3 examples here. Okay? Nereis, Pheretima, Hirudinaria. Nereis, Pheretima, Hirudinaria. What's that? It is Nereis, Pheretima, Hirudinaria. Nereis, Pheretima and Hirudinaria. Okay? As I said, for the first time you will see the segmentation here. So you know that when you talk about the external segments, you talk about the external segment. These are annulus. This is annulus. Plural is annuli. These groove-like structure which are dividing the body externally. And internally, I told you what will be there. Internally, I told you what will be there. What will be there? There will be septa. There will be septum. Septa is there, and nephridia will be repeated. Okay, nephridia will be repeated. Nephridia will be repeated here, right? The repetition of nephridia will be there. Okay. Okay. So here, and even you will see closed type of circulatory system. Closed type of circulatory system will be there. Now, it's a non-chordate, right? The one which are not having the notochord, they all comes under the category of non-chordates. Okay? This Annelida is also a non-chordate. But in the case of non-chordates, keep one thing in your mind. Just say in our case, we are chordates. In our case, the notochord, right? The notochord is replaced by vertebral column. So basically in our case, that notochord is making that vertebral column. Yeah, it is replaced by vertebral column. Our heart is on the ventral side. This is our dorsal side. This is our ventral side. These are our lateral sides. So our heart is on the ventral side. So if in non-chordates, if heart is present, if heart is present, it is on the dorsal side. Always remember that. If they have the nerve cord, right, that nerve cord will be on the ventral side, and it will be solid, it will be double. Solid matlab jaise in our case the nerve cord is hollow, it is single. Jaise bamboo is hollow. So here if nerve cord is present in non-chordates, it will be ventral, it will be solid, it will be double. Okay, it will be double. This part clear? This part clear? So if heart is present, it will be on the dorsal side. If nerve cord present, it will be on the ventral side, it will be solid, it will be double. Do remember this. Do remember this. Okay, this is important. Okay, it is important. Done. So for the first time, right, you will see the segmentation here. They are coelomates. Okay, this part is done. Okay, so here you will see the head formation will start. Matlab, even in platyhelminthes, you won't see a proper head. Matlab, that's, see, head formation is known as cephalization. Head formation is known as cephalization. What is it? Cephalization, head formation. So you are right. You are not going to see, like in the case of Annelida, you will see anterior head is there having sense organs, right? But in other cases, you won't see a proper head development is there. You can say that the cephalization has started a bit, but it is not, right? The proper head formation is not there. Even in Annelida, but in Annelida, you will see that anterior side will have the head that will contain the sense organs, right? Anterior side will have the head that will contain the sense organs. Is that clear? Is that clear? Yes, is that clear? So when you talk about their body, so body wall mein cuticle, epidermis and muscular layers will be there. Okay, what will you see? You will see cuticle, epidermis, and again muscular layer will be there, right? Muscular layer will be there. So cuticle is moist. Cuticle is what? Cuticle is moist. Cuticle is what? Cuticle is moist. It is elastic and elastic. And obviously you know that gaseous exchange will be there directly from the surface. Then epidermis is the word, right? So it is living, but it will secrete this cuticle. Okay, bache? Okay, bache? And muscles mein you will see both types of muscles are there. So it is living and secrete cuticle above it. And muscles mein you will see longitudinal and circular muscles are present. Longitudinal and circular muscles are present. They are present. Is that clear? They are present. Is that clear? So longitudinal and circular muscles are there. Just in our case, it is ICOL. Matlab, in our case, we have inner circulatory muscle, inner circular muscle, outer longitudinal. Right? I'm talking about myself. I'm talking about the humans. We have inner. Our inner muscle is circular. Our outer muscle is longitudinal. But here you will see that inner muscle will be your longitudinal. Outer will be circular. Outer will be circular. Okay. But muscle layers are definitely present. Muscle layers are definitely present. And these muscles, bache, these layers, it will help in locomotion. Okay. It will help in locomotion. It will help in locomotion. Is that clear? Is that clear? Now they have some setae and some appendages for locomotion. Matlab, in this particular phylum already you will see the segmentation is there. You will see they are coelomates also. So here you will see locomotion. So when you talk about the locomotion, you will see chitinous. Yes, chitinous setae. They are S-shaped setae. Right. We'll see it in the case of earthworm. Right, and the lateral appendages. And the lateral appendages. What do we see there? We see the lateral appendages, right? That is parapodia. Parapodia, and it is in the case of Nereus. It is in the case of Nereus. So what you need to remember that in earthworm, it is having S-shaped chitinous setae, right? It is having S-shaped chitinous setae, bache, and that muscles will also help in the movement. And here this parapodia, right? Let's say this is their body. right? Lateral side will have some appendages, that is parapodia in the case of Nereis. Clear bache? So this Nereis is also known as yes, it is also known as sandworm, right? It is also known as sandworm. Is that clear? Is that clear? Right, it is also known as sandworm, right? I think I somewhere I mentioned rugworm, na? Rugworm is there, I need to check it again, but it is sandworm. Clamworm or sandworm, that type of word is there, you go for the sandworm, okay? Also known as sandworm, okay? Okay, so in the case of Nereis, you will see parapodia. Now, bache, Nereis is what? It is dioecious. It is dioecious, okay? So you will see that in its reproductive stage, right, you will see how its structure will change, right? So it acquires a form during this reproduction part, heteronereis, right? Heteronereis will form. Do you know about this heteronereis? Yes. Do you know about this heteronereis? Yes, please. Yes, please. Heteronereis, do you know anything about this heteronereis? So, bache, it's a free living dimorphic sexual individual, right? It is a free living dimorphic sexual individual. It will be there on the surface of the sea, right, bache? It will be there on the surface of the sea. Clear, bache? Okay. Yes. Okay. So when you are saying Nereis, it is sexually immature form. But when we are saying heteronereis, it is the sexually mature form. Right? Sexually dimorphic form it is. Yes, they are the true worms. Right? They are the segmented worms rather. Right? They are the segmented worms. What is the word that we use for Annelida? One student is asking it now. They are known as segmented worms. Okay. They are known as segmented worms. Okay, so they have parapodia. Now in the case of leech, no chitinous setae, no parapodia. Please remember this. No chitinous setae, no parapodia. Do remember it. No chitinous setae, no parapodia. Okay, so ye dekho the segmentation here in their body, right? So see, internally it is septum. Internally it is septum. Okay, it is having these tube like structures. Okay, bache, and outer groove is there. Okay, and see this leech. See this, it is leech hirudinaria, right? It is leech, it is hirudinaria. It is hirudin. Why do we say, see, from this leech hirudinaria, it is having the suckers, right? On both the sides it is having the suckers. But if from this hirudinaria is having hirudin, what is it? It is natural anti-blood coagulant. It is natural anti-blood coagulant. It will not, just we have heparin, here you will see hirudin is there. And look at this Nereis, sexually immature, and heteronereis is sexually mature. Okay? Okay? Yes. Any doubt? Any doubt? So leech is a, yes, blood sucking parasite. It's a blood sucking parasite. What's that? It is a blood sucking parasite. So we use the word sanguivorous for it, right? Those organisms who feed on blood, sanguivorous. Those organisms who feed on blood, sanguivorous. Right, bache? What are they? They are sanguivorous. Clear? They are sanguivorous. Clear, bache? Clear. So ye dekho Nereis. Okay, then parapodia. Okay, so that's what we have. This is the Nereis. So see the lateral appendages. They are parapodia. Fine? They are parapodia. Chaliye, so this is all about the phylum Annelida. So they may be aquatic or terrestrial or free living, and some are parasitic, just your leech. Okay, They exhibit organ system level of organization and bilateral symmetry. They are triploblastic, metamerically segmented, and coelomate. Simple. They have the segments also known as metamers. So Annelida. In Latin, annulus means little rings, that grooves that are, that is showing the external segmentation. So they have longitudinal and circular muscles which help in locomotion. Important it is. Baki bache lateral appendages done, and for excretion they have nephridia. This is also important for excretion and osmoregulation. This is not protonephridia, this is nephridia here. This is nephridia here. Baki here also bache neural system will have paired ganglia, right, connected by lateral nerves to a double ventral nerve cord. So do not forget this. They are non-chordates. So nerve cord is on the ventral side. It is double. It is solid. Okay. So examples be done. Next is arthropoda that we will start after a break. Okay. Let's meet at 10:40 p.m. Okay. Chalo. Then let's meet. Fine.